as there are multiple versions. So that is the reason I have shared the FragPipe JRE 20.0.zip. So the simple thing is that you have to just download this .zip file and unzip it. That's all. So you don't need to download any extension from the internet. So I hope that this much is done, right? Right? How many? Can you please raise your hand? Okay, so you guys have downloaded this one, this file, right? And we need the control one and treatment one. So we are letting you know beforehand. The reason is that the file sizes are almost 1.7 GB each. So that is the reason you have to, we have asked you to already download these files. Otherwise, executing the software will be difficult. And one more files you have to download, which I have already told, that is the output combined file. So this particular file will get generated after analyzing control and treatment using FragPipe. Okay, so I believe that if all of you have downloaded at least the the zip file. So then you will see after unzipping, you will see this particular file folder, which is a FragPipe folder. Once you open this particular folder, you will see multiple folder. In that, you will see a bin folder. Once you open in the bin folder, you will find this application, FragPipe. Double click that, and this particular GUI interface will open. Can you please tell me that how many of you has reached to this? Okay, few of you. Okay. And what about others? Like, why are you guys facing any issues? Anybody are facing any issues in downloading the files, or because I think reaching till this will not be a difficult part. What? Can you? Yeah, in WhatsApp will give. But again, I think Ankit will be forwarded. Okay, there are a few small steps are there. So let me just tell, hoping that most of you has reached to this particular page. The first thing is that as we will be doing the LFQ analysis today, so for that we will be requiring few of the tools to download and update. Okay, so first of all, this is FragPipe. FragPipe is the software. MS Fragger is the search software over there. So for that particular thing, first thing is that you can see that there is MS Fragger. Can you see? So in this MS Fragger, you have to come over here and click download and update. So once you are clicking download and update, you will see that it is asking for three some information like your name, institution, and a mail ID. And you will have a series of 4 to 5 checks that you have to check in, and then it will start downloading. It varies from PC to PC, but it will take 2 to 3 minutes for downloading the whole MS Fragger. So that you should do it right now. Second thing is that we will be requiring ion quant. So for that, the same way, once this is done, you can come to the download and updates. Same way, it will ask for some information. But the memory of trackpad is pretty good. Then it will remember all your information from the MS tracker and it will help you to download. The third is that philosopher. So it's generally for the calculator and the database. Again, you can come and download and update. This time it will be a bit different. It will show either automatically or manually. So you will check automatically and it will complete the download. For this session, we don't need the Python. So it's fine if you are not downloading it. But if you have the extension in the PC already, you can just relocate or just give the path over there. And you can by clicking on the browse or you can download, but it's not required. So before we start the analysis at 10:30, make sure that all these tools are downloaded, configured, and we are in this particular page, which should show these, this particular path over here, and also the versions over here. Otherwise, we will not be able to change any parameters when we are moving into the workflow or over here the MS tracker or into the quant. So that should be taken into consideration. So we will have some of our teammates like Devargo, Shaurami, and Suraj, who will be helping you during that time if you are facing any issues regarding the analysis. But they will also not able to help you if you guys are not reaching to this particular page. So that particular, reaching to this particular page is important. Okay, so we have 4-5 minutes more. If you have any questions, any queries regarding reaching to this particular track pipe page, please let me know. Like from the net, I downloaded all the three. So I can give the path on that and use it, right? Yeah. So you have downloaded all these extensions, like manually, and you are putting it. That is fine, not an issue. But sometimes, like for example, the versions, you should check that if we, download and update it from here, the version that it will take is the most, like the updated version. At the same time, if a newer version is coming, it might happen that the particular newer version don't work with the track pipe. So they generally knows that, and accordingly they put the path in such a way that it will download only that particular version, the version which will help you to, which will work and sync with track pipe. So I recommend that what he's telling it's another approach where we can go to each of these tools, because these are all independent tools, has been put together under TrackPipe. So which can be those particular tools can be downloaded separately. You can put all these tools in the C drive of your PC, and you can browse that using this particular icon, and you can give the path. So that is another way if some of you are facing any issues in downloading and updating. Any more questions? Okay, I think few of you have just come. So just briefing you that this particular drive we have shared yesterday. Please make sure that you are downloading the control1.raw, which is here, treatment1.raw, which is here. So these are the two MS raw files that we are providing. Then control2, control3, raw treatment2, treatment3, we have given it so that if you want to try it with more number of samples, pre-control3 treatment, you can do it afterwards. But today, to expedite the process, we will only show the analysis with two samples, control one and treatment one. If things goes fine, according to our experience, it will take 15 minutes to run the whole analysis in PrepPipe. So this is MS Robert. Excel? No. So it requires mass spectrometry generator drawbell. It can be, we have given from thermo electro Orbitrap. Thermo Orbitrap. However, if you want, you can use any other extension that if you have some files. But as of now, just to give the demo, we have given these raw files. Okay, these are the two files that you should download. Third is the pracpipe.zip. This is the main application which we have requested you to download beforehand from the internet. But as you guys are facing some issue, so we have got this particular .zip, which is the current version. And as of now, we are not facing any issues with this current version. So this one you should download, unzip it, and you will see a folder which is PracPipe. This is the folder you will see. Once you are unzipping that, click this one. Then you will get a folder which name is bin. Under bin, you will get an application folder, PracPipe. So this should not give you any kind of Java related issues that many of you are facing. The reason is that in this particular folder, like zip files, we have already uploaded the extension. That is the .jar extension. So that should not give you any issues. But still, if you are facing any issues right now, please let us know that we can come and help you. But these files should be downloaded beforehand. Otherwise, you will not be able to follow the steps over there. Okay, so apart from this, I will also request you to download the output file. So this is the same output file that we have run yesterday with the control one and treatment one, so that we can help you to do the analysis, at least to show the data, even if the run is not getting over. Okay, so we have a minute more. So if you have any questions, doubts, queries, please let us know. Okay. Yeah. So this is the PracPipe interface. Once you are opening that particular application, make sure you are downloading and updating at least the MS Fragger, which is the search engine, and Quant, and Philosopher. No need of Python right now. So these three should be downloaded and uploaded. You just need to click over there. There are few information you need to add, and it will automatically download and update. For Philosopher, there will be two options. One is manual and another is automatic. You guys should choose automatic. Okay, I think that's all from my end. I think now I think whatever is required, let us know.
You can move around it, that will be best.
Yes.
Yeah, so I think the QPO Congress stands out because this I think is one of the challenges in organising the QPO Congress, in that it covers a broad spectrum. That it covers both the technology and the detail of the technology. It covers the applications of those in the research laboratory, and increasingly it's covering the translation of those to the clinical setting. QPO President, what are my goals for the next 5 years? Well, what we'd like to do is many things. We want to keep advancing the field, advancing technology. We also want to keep making fundamental discoveries in human biology. But I would argue it's more than that. We really want to also interface with the community and get proteomics out to a larger group.
Just over a year and a ago, as Vice President of the United States, Obama put me in charge of what we refer to as the cancer moonshine. Our effort to put together a strategy to end cancer as we know today. And we had a line in the cure of Troy that I think best characterizes where we are today. And it goes like this. He said, history teaches us not to hope on this side of the grave. But then once in a lifetime, a tidal wave of justice rises up. Hope and history crime. And I can't think of any more prominent group of people who may be responsible for making hope and history crime than all of you in this room today.
Kind of look at that. I want to have the scientists and representatives from industry, which are really benefit from proteomics.
We are helping us with time and take this more time. Thank you for your. We're also, it's also very good way of keeping up.
The first time to clean up proteomics laboratory expertise with an ongoing NCI-sponsored clinical trial.
Opportunities created by scientific wellness in a healthcare that is predictive, preventive, personalized, and participatory. How do we bring them together in a coherent fashion? And that's what I hope this Congress and its leadership can attack for the future. Can you articulate a vision that places proteomics in the main flux of this revolution in medicine?
My boss, the students.
Thank you.
After spending my formative years at Harvard Medical School, I was clear that I needed to come back and set up a world-class proteomics laboratory here in India. IIT provided me with an ideal platform. It's a kind of stepping stone, making my start quite visible. My family took a bike to Harvard to the new surroundings, but then IIT was such clinical research requires taking up. In the initial months, I focused on obtaining competitive grants from the various governmental agencies. As expected, these funds took time to materialize. Thus, Proteomics was the new medical country, and the funding agency had their position.
अभी तक मास स्पेक ओके तो ये तो फोन
Like students with like international labs, I want my biomedical students to have support of various analytical tools. These belong to the
आप लोग इधर से दिखा दो
I outsource them from interdisciplinary background, like mechanical engineering, and even aeronautics.
I'm looking to study the effects of microalgae on proteomics. The big picture idea is that we should be able to develop applications that will be even beneficial to absolute energy.
These students could easily do complex data analysis. Everything was now in
ছোট সবাই বাইরে all the four cohorts have to be compared simultaneously. This is our data analysis platform. চেষ্টা করছি কিন্তু কি দিতে বলে স্পিড নিচ্ছে
just before my tension doctor taking work and finding success in getting the government
আচ্ছা আমি অফিস থেকে জিজ্ঞেস করে তোরা কিছু কানেক্ট করেছিস ওয়্যারলেস তোরা ওয়্যারলেস
more funds loaded thereafter. My lab started purchasing the state-of-the-art equipment one by one. The journey has taken a turn. With such resources at my disposal, I could now focus on setting up the protocol for clinical testing. My students were gradually introduced to ethics and philosophy of working on clinical samples. Shoot of the video advanced instrumentation and systems brought in recognition at the global level. But it's possible now. They're going in rockets. In America, my friends, it's the last time. How has been the journey? In the orbit. Just my
Hello. So there is one announcement. If you are not finding the Wi-Fi, go to the IITB guest ID and you log in with the Wi-Fi and the password that have been given into WhatsApp. Okay, go to the IITB wireless guest. Your account.
Around something?
Your account will now. The high resolution transcript of the treatments will be used to subject to the facility. IIT Bombay. Even covered high resolution Indian research community with the help of state-of-the-art proteomics instrumentation. The facility aims at offering services ranging from proteomic and metabolic investigations of samples, understanding pH in complex samples, validating target proteins and metabolites, and their relevant and functions in the application. Community can also benefit from access to other platforms available at IIT Bombay.
Hello. If there is any issue with IITB guest regarding the speed issue, then please log into IITB wireless with the same ID and password.
Your problem. Ladies and gentlemen, welcome to the workshop. Our next session is about observation. And I request you to kindly mute your microphones and wait as we prepare for the session. Thank you.
Should be something.
Ladies and gentlemen, welcome to the workshop. Our next session is about observation. And I request you to kindly mute your microphones and wait as we prepare for the session. Thank you.
All right, good morning, everyone. Welcome back to the day 2 of Monsoon Advanced Proteomics School. Looks like you're all very much enthusiastic and prepared already today for the hands-on sessions. So from the last day, we had few activity which we have started yesterday, right? You got the quantification values for the protein quantification which you have done. We hope you have already made plots. If not during any break, please do that. And if any problem, let us know. Additionally, you have done the gel, which we will see how that came today sometime afternoon. We had given you 2 demonstrations of instrumentation, different mass spec, as well as cell surface proteomics protocol. And then at the end, we talked about in solution digestion, which now considered for the overnight done. Now those samples are ready to be injected in the mass spectrometer. So the plan for today is we are going to first have a lecture right now from Dr. Santosh Renusle from Thermo Fisher Scientific from California. We will talk to you about one of the most recent innovation in orbitrap technology or mass spec technology per se, the Astral Orbitrap, which is pretty much sought after the technology right now. So we will hear his talk on the deeper proteome coverage, how to obtain that with the Astral Orbitrap. But the next session will be what I'm sure you are waiting for, how to analyze the mass spectrometry data yourself. But what we are going to do more innovatively, we are also going to show you how to run the samples live on the mass spec. So your one sample we will be injecting and we'll be showing you here how that data comes, like how you are acquiring chromatogram. And of course, we are providing you the mass spec file to practice yourself. We will use the MS Tracker, FracPipe software, which is one of the recent one in the field, but very user-friendly, very fast. So that Deep Biswas and the team will guide you through. We will have a very short demo after the tea break, which will be a new kit, Proteomics, which I think is very important to do in India now. And this is the first time they are going to show here. I'll talk briefly about the new field emerging single cell proteome. And then we'll have a detailed session on the single cell proteomics. The single cell field is right now evolving very much, but where we have made the maximum impact is about single cell transcriptomics. Proteomics is just following right now. So Dr. Saket is going to give a short lecture followed by a hands-on session on how to analyze the data of single cell transcriptomics. A lot of hands-on activity today. Then finally we will have another session from Professor Graham on the looking at machine learning approaches for biomarker discovery. So throughout, you have to be attentive. Things will go fast. A lot of hands-on activity, but I'm sure this is going to be very useful for you to take these knowledge back with you. And I'm sure this one day may not be sufficient to imbibe everything, but you will take some time. But please make sure that we're going to have our first lecture from Dr. Santosh Srinivas, and Sachin will introduce him, and then we'll have his lecture.
Good morning, everyone. So we now have the first speaker for today, Dr. Santosh, who is a vertical marketing official scientific. Dr. Renews obtained his PhD degree from the Institute of Bioinformatics in Bangalore, India. His expertise is in field of applying mass spectrometry-based proteomics and phosphoproteomics for biomarker discovery and identification of therapeutic targets. Dr. Renews has worked in the field of clinical proteomics to optimize LCMS methods for development of clinical.
Are you able to hear me?
Yeah, good morning, Dr. Santosh. I think we can get started now.
Hey, good morning, everyone. I hope you must be enjoying the comprehensive workshop that Dr. Srivastava is conducting here. I've been very fortunate to be able to participate previously from Thermo at India. I'm again, thank you, Dr. Professor Srivastava, for providing me this opportunity to present our new Orbitrap Astral mass spectrometry. So today I'm going to talk about the newest mass spectrometer from Thermo that we launched at American Society for Mass Spectrometry conference couple of months back nearly. So it's about a deeper protein coverage and faster throughput for bulk and low input samples on Orbitrap Astral mass spectrometer. So let's think about the central dogma of biology in general. So most of you probably aware of that. Central dogma runs through DNA or RNA. So whole genome sequencing have been developed pretty comprehensively nowadays. Then you can have entire genome sequenced and get features of all the genes across the organisms. And then we have the second is transcriptomics, which provides insight into the expression patterns of all the genes that these genes encode. And then now, at the end of the day, we have these proteomes, which are functional units of the cells and organisms that basically does all the work. So at the end of the day, proteomics is very important to study, the functions of the cells. But why this proteome is analysis of proteomes is why so difficult? There are so many analytical challenges. Probably must be discussed at the current workshop. So there are higher dynamic range in terms of proteins, several orders of magnitudes. Abundance differences happens in proteome, especially in terms of plasma. You have 10 orders of magnitude difference between the abundant proteins and the lowest abundant protein in the same sample. That's the low abundance challenge, especially important in terms of single cell proteomics, where the sample amount and protein amount is very limited. Apart from that, there are so many proteoforms that are present due to post-translation modifications, be it glycosylation, phosphorylation, so on and so forth. The complexity of the proteomes is increased drastically due to these proteoforms. And again, the protein's expression varies depending on the time and space during each and every cell has a different expression pattern of the proteins. And that adds the challenges in studying these proteins. So overall, we think that the promise of proteomics has yet to be fully realized due to current generation analysis technologies. Now, these are the major challenges that we face today. That we do not have any sufficient throughput to analyze thousands of samples for meaningful biological analysis. And most of this analysis basically includes not all analytes are present in all the samples, and there are so many missing values that we need to deal with, and that's why we will not be able to study these proteins effectively across multiple different samples. Again, the lack of sensitivity to be able to detect the low abundant proteins are ever increasing, and we are not able to detect samples which are limited in protein amounts, such as single cell protein samples. So to address these important questions, we launched this very advanced Orbitrap Astral mass spectrometer. Astral meaning for asymmetric track lossless ion mass spectrometer. This is a schematic of the mass spectrometer. Through this mass spectrometer, we basically achieve faster throughput, deeper coverage, and higher sensitivity. And at the same time, we basically achieve accurate and precise quantitation. How we do that? I'll just go through those data. So first of all, faster throughput. In order to analyze and get meaningful biological insight into the samples or diseases, we need to analyze many different samples. So that in current mass spectrometries, we cannot do that. We have basically achieved maybe 48 to 50 samples per day analysis, and it will take at least a month or two to be analyze thousands of samples. So to address this, we have new mass spectrometer which can analyze 180 samples per day with a deeper coverage of the proteome. With this proteome coverage, we can get up to like more than 8,000 proteins identified in less than 8 minutes, around 8 minute samples to sample injection time. So if you think about it, in the current generation mass spectrometers, we can analyze one sample in 30 minutes. So in the same time, we can analyze 4 samples using Orbitrap Astral mass spectrometry. That's 4 times faster than current generation mass spectrometers. So through that, let's consider 13-hour time period where 26 samples can be analyzed, but now we can analyze full 96 well plate in the same time frame. So basically within 24 hours, you can analyze 296 well plates with Orbitrap Astral mass spectrometer. We can get deeper coverage. So nearly we today with respect to multi-dimensional chromatography, we can identify up to 12,000 proteins, but this requires higher protein amounts to start with. Then also requires to do pre-fractionation or offline fractionation, collecting those fraction, concatenation of those fractions, and then analyzing each of those fractions onto the mass spectrometry, which takes roughly about a day or two of mass spectrometry time. All this cumbersome process needs to be routinely taken care of. And also there are a lot of variation and quantitation becomes challenging in this kind of samples. Now, in the same time frame, we can identify more proteins. So in this case, we can identify more than 12,000 proteins in about 1 hour time. If you want to go deeper, you can spend more time on mass spectrometry. In this case, roughly about 4.5 hours, you can identify 15,000 proteins, or else you can do the regular methods which are faster. Up to 30 samples per day, you can identify up to 10,000 plus proteins. Now we have basically ran multiple different sample throughput methods with using 200 nanogram HeLa digest from 300 samples per day all the way to 30 samples per day. In this case, in 30 samples per day method, we can identify up to 7,700 proteins. And in 30 samples per day method, we can identify more than 10,000 proteins in technical triplicate samples of HeLa digests. So further, we applied this workflow, end-to-end workflow for a cancer cell line, set of cancer cell lines. So in this case, we used acceleron, which is automated sample preparation, which can process 36 label-free samples in about 6 hours from slicer to peptide digest. We used the Vanquish Neo AU HPLC system for for separation of the peptides using micropack Neo column on an Orbitrap Astral mass spectrometer using a DI methods. In this case, we used 1 hour methods for analysis of cancer cell lines, and we analyzed the data using Proteome Discoverer software inbuilt with Chimeris database DI data analysis search engine. We used following 11 different cancer cell lines using the acceleron platform, and we achieved a higher depth in each of these biological triplicate samples, those which were analyzed across 3 different days. Up to 9,000 to 10,000 proteins were identified per cancer cell line. This also gives you a very high precision. The CVs were less than 15% for most of the cell lines that were studied in this case. In addition to cell assets, we also did deep plasma proteomics analysis for plasma kind of samples. It is very challenging, and current generation mass spec can identify a few hundred proteins in about 1 hour mass spectrometry analysis. Our mass spectrometry with Orbitrap Astral DI analysis using 180 samples per day method, we were able to identify more than 600 proteins. With 14 samples per day method, we were able to identify more than 1,100 proteins using neat plasma. When we enriched the plasma using a shear nanoparticles approach, using 5 individual fractions, approximately 36 samples per day, we were able to identify more than 3,100 proteins. Those fractions, if we pull together and run one long gradient of 80 minutes per full fraction, we were able to identify more than 5,000 proteins. If you want to go even deeper, each of those 5 individual fractions were analyzed using 5 long gradients with a throughput of 4 samples per day, you could achieve more than 6,300 proteins in the same plasma. The plasma amount was loaded is about 500 nanogram. So as you can see, we have achieved throughput also coverage, and sensitivity is also equally important to be able to detect lower abundant proteins in such samples. So digging deeper into the protein becomes extremely harder the deeper you go. In this case, for single cell kind of samples, we would like to have a higher sensitivity to be able to detect those low abundant proteins. So in the current generation mass spectrometers, we can identify up to 3,500 proteins from 250 picogram lysate using library-based approach. Now with Orbitrap Astral mass spectrometer, we can identify up to 6,000 proteins using twice the throughput. So previously we used to do 40 samples per day for identification of 3,500 proteins, but now with 80 samples per day method from 250 picogram HeLa digest, we can identify up to 6,000 proteins. We further analyzed the HeLa digest dilution series from 50 picogram to 10 nanogram. In this case, using library-free approach, we will identify for 250 picogram, which is about 1 HeLa cell amount, about 4,000 proteins were identified. And for 10 nanogram digest, we will identify up to 6,700 proteins. When you use that library-based search using a library generated using 10 nanogram HeLa digest triplicate runs, we were able to identify up to 6,000 proteins from 250 picogram HeLa digest. Those proteins basically can be identified at higher levels using specific library rather than a bulk library generated from highly fractionated samples. Equally, it is precision of quantification is equally important, as I mentioned earlier. Overall, we were able to identify more than 75 identified proteins with less than 20% CVs, and about 50% of the proteins were less than 10% CVs across the dilution series. We also analyzed HeLa single cells that were processed using Selenium system from Selenium. In 12 replicates of HeLa single cells, we were able to identify 3,200 proteins using library-free approach, or more than 4,300 proteins using a DI library-based approach. The DI library was generated using 20 cells and 6 cells at HeLa digest runs using Spectrum 18 software. In this case, with respect to throughput, deeper coverage, and sensitivity, accurate and precise quantification is equally important. So we analyzed 3 proteomic samples. In this case, 500 nanogram of 3 proteomic samples were analyzed on mass spectrometer. 250 nanogram was human protein, constant across the different mixes, and E. coli and yeast were varied as mentioned here. So we have been able to show that even if we have 1:9 or 1:1.5, the proteins were accurately and precisely quantified, and the expected ratios were shown here. In this case, the yeast expected ratio was 1:2:1, and we were able to get the ratio of 2.04. In terms of E. coli, in the case, the expected ratio was 0.25 of 1:4, and we were able to identify 0.21. It's very close to the expected ratios. Also, as I showed previously for the single cell, for bulk cells, more than close to 90% of the identified proteins were less than 20% CV, and more than close to 75% of the identified proteins were less than 10% CVs in this label-free kind of benchmarking sample. Now, let's go a little deep into how the Orbitrap Astral mass spectrometer looks like. So here you can see the schematic of Orbitrap Astral mass spectrometers. There is a quadruple mass analyzer, and then there is a high field thermoscientific Orbitrap mass analyzer, and this is novel Orbitrap Astral mass analyzer. So the quadruple analyzers has high transmission rate. You can isolate close to 0.4 Dalton isolation window. The ion isolation switching times have been improved. It is close to 1 milliseconds on this improved quadrupole on the Orbitrap Astral mass spectrometer. Other than that, we have the brightest ion source in the industry with the letter box kind of ion source, high for high transmission. Then there is advanced ion beam guide to reduce filter ions and reduce the neutrals going to the mass spectrometer. The ultra high field Orbitrap mass analyzers can get scanned up to 480,000 resolution at the scan speed of 40 Hertz. It has a high dynamic range up to 1:5,000 and mass accuracy at parts per billion level. The novel Astral analyzer includes ion processor for peptide fragmentation. Then there is injection optics to inject the ions into the Orbitrap analyzer. All the parts of Astral analyzer, ion foil and asymmetric ion mirrors basically gets a track up more than 30 meters in length. The travel time between injection optics to detector here is less than 5 milliseconds for every ion packet. Now I can show you the different high resolution DI scan that happens in Orbitrap Astral mass spectrometers. So when you inject the ions for full scan, the ions are basically collected in ion routing multipole using AGC and maximum injection time. Those were then injected into the Orbitrap for detection. Once the full scan is happening, the second set of ions are basically isolated in quadrupole and enriched in ion routing multipole. Those ions were then put into ion processor for fragmentation, and then they're injected into the Orbitrap Astral analyzer and finally detected in high dynamic range detector. So with Orbitrap Astral, we have basically worked towards parallelization of different ion packets throughout the mass spectrometer. So we can have process 5 ion packets simultaneously. First, the Orbitrap full scan happens at 240K resolution. Then the second set of ions basically comes in ion routing multipole, fragmented in ion processor, and then it injected into the Astral analyzer. And simultaneously, as you can see, there are 5 different ion packets which are processed at the same time, which basically gives high scan rate of up to 200 Hertz. So as you can see here, you can have 240K Orbitrap resolution scan in 0.5 seconds. That's the transition time required for 240K resolution in Orbitrap. In the same time frame, the Astral analyzer can scan for 100 MSMS fragments in the Astral analyzer at the 200 scan rate. Overall, we have end-to-end solution from sample preparation from acceleron to LC separation, column chromatographies, FAMES for ion filtering, and better increase the signal to noise as an ion mobility device, and Orbitrap Astral for high speed, for higher accuracy, faster throughput, and deeper coverage, and higher and better quantitation Orbitrap Astral mass spectrometer. And the new solution that I didn't talk about is the RDF for data processing. So overall, we think that with this new mass spectrometer, there are several possibilities that is now available for all the scientists, and we basically ask everything what is possible with the new software and technologies. Thank you. I can take any questions if you have.
All right, Dr. Santosh, very nice lecture.
Thank you.
You can also see the audience here. All right, any questions from the audience? So while they're thinking about some questions from you, so let me appreciate like how these technologies are truly transforming the proteomics. What happened here? Yeah, you know, the way now we are seeing the revolution happening. In fact, you know, from single cell, we were talking few hundred proteins earlier. Then it reached 3,000 or so with the timsTOF, and now I think with Astral, you're showing 5,000 plus data, which I was amazed to see. And I must say to all the participants, we're at a very lucky time of proteomics. You know, when every day there is so much competition, and we are not talking to the magnitude which was never achievable earlier in the past, right? So with now these new instruments, new mass specs, your entire biology can be very much transformative in a very short time. So great. Any questions from the audience? Oh no. Okay, a very practical question about price, right? So the price will be quite huge.
So yeah, my best answer is I'm not the correct person to answer the question. That question. Yeah, maybe you can reach out to me. I will guide you to the correct person. You know, that way.
Right. So by the way, I just want to tell the audience here, like, you know, next year we're going to conduct a conference in 2024 February. I will tell you later on. But in that conference, one very distinguished person is going to come. His name is Alexander Macraw. He's the person who made the Orbitrap technology and now the new launch of Astral Orbitrap. So he's the person who actually invented these technologies. And we are very fortunate to have him in person to listen about his innovations and share his knowledge with all of you. So that will be something to look for. But yeah, Santosh, thanks so much for sharing this knowledge. And it was amazing to see. I'm sure the numbers are too, difficult for our audience to digest so easily. You know, these are all magnitude of 10,000 plus and those numbers. But we are very much looking forward how best we could also start using the Astral in India. You know. And but it's like really revolutionary. I'm very happy that how proteomics is transformative right now. And what we saw in 2005 has actually now, maybe 17 years later, it has totally now got a big change again. So let's see how it pans out. Great. All right. Thank you very much for waking late in California and giving this lecture. Thank you once again, everyone.
Thank you. Thank you for the opportunity. Yeah. Have a nice time.
Great. All right. So just continuing on the session now, what we are planning to do, we are going to show a live mass spec run from the lab. Let me see if Surbhi and Avinash, the TAs, can actually share the screen and show you, injecting the mass spec sample peptide. Good, they're ready. What we're actually thinking that we will keep one window open where you can see how the run is progressing. So even if you were to stand in front of mass spec, you can't do anything, right? You will simply see how the chromatogram is progressing in 1 hour or 2 hours of gradient. So Surbhi, I think you have just started already this run, right?
Yes, sir.
And can you please now give overview what sample you're running, what gradient, and then you can take over, show the settings for mass spec and LC.
Okay, sir. So we'll share our screen. So I have shared my screen. I hope it is visible.
You have to, zoom into the right frame. So.
Ankit, I'm not able to share my screen. Is my screen visible? No?
or MGF or any of those mass spec for file, which you are going to analyze today. So what you will be given is the actual raw data file, which Deep is now going to take over, and how to process that file. In the previous workshop, whenever we have conducted, given that the amount of the time it takes to analyze the data, we were not able to give the raw files to the participants. We used to give the processed Excel file. But now this MS Friday software has become so fast that now you can do it in the real-time manner. So that's why we are taking risk and giving you the raw file. Now what this actually means that now you have full ability that you can send a sample to anywhere, even if you don't have mass spec in your lab, to any facility, and ask them to give you raw file. And now you have full control on your data, right? How you analyze the data. And you will see that now you can make the best use of your abilities to do learn proteomics and get the maximum output from the data file. There is no one best database available. You will have to try many things. There is no one best filtering criteria or the different discriminancy parameter. So now you will see how you can play these things yourself, right? So it's a good idea for you to pick up this part. And I'm sure now as you will go back from this workshop, you will be in a full commanding position of taking the raw mass spec file and analyze yourself. Yes.
Can you, sir, can you just explain about the starting part? If the troubleshooting has to be done, because this is a smooth error. But
A lot of troubleshooting happens, right? A lot. Many times, let's say the peptide which we are injecting contains impurities, contains salt. The very first thing which we are always afraid of when you are injecting a new peptide sample, it may even start showing bubbling. The column may get clogged if the peptide was not cleaned properly. And believe me, this is the best people who are doing proteomics in the lab. Is still very often we see this kind of issue. So we have to be extra careful of cleaning these samples properly. And your injection has to be you don't want to overload the peptide either. A good recommendation is going to be 500 to 750 nanogram, maybe up to 1 microgram. But just if you want to improve coverage, you should not go to a 5 microgram of peptide. It will again, clog the column. So these are the, nano LC what we're talking, right? And very minute capacities. So if any impurities, any overloading happens, that the first culprit happens. Second thing, of course, what we are running is we have optimized the gradient for it. We know that, okay, what will be best gradient to get the Gaussian pattern. But you have to decide, okay, like maybe at 30% or 45%, how long I want to keep. Okay. And then I want to keep it continued for a longer time, then I'm going to increase 85 or 90% to get everything out. So again, that peak optimization. So in the beginning of any project, where let's say you have 20 samples to work with, it's good idea to take a pool of it, optimize the conditions, right? And LC parameter is becomes one of those. I think they will show shortly the LC parameter. And likewise the mass spec parameters, right? In the MS level, what resolution you should use, what age you should use. Again, different. So we'll not bias you what is compared. Use of your setting. Different setting. Somebody uses size in different setting. So like we don't want to go to the very specific meaning of the word thing, but in general, optimize what is best resolution. Best target, how much time to do for the MS fragmentation, all of these in document. Once 1 or 2 good runs has happened, then you are good to go for 20, 30, 40 or 100 samples. Like, yes, we have a question. But that's right. So that's what I'm talking. But I said, like, what we will show you is the NSMS parameter for the thermal instruments, right? Which is very commonly used a lot of places. But that should not be the only thing you should be knowing, because again, the Bruker or SciEx or Agilent, they will have different meaning or different wording, but concept will almost remain same, right? You'd like to learn those stuff. So I think let's go to the actual reality of the software interface, and they will quickly walk you through the LC and MS parameters. Can you to share the screen now? Don't share the MS. Okay. Let them just take a minute. Then I think. Yes. LC parameters. Okay. Then I get them. Windows is share codes. Okay, our idea is while you are all sitting here, you should definitely feel confident that you are doing this on yourself, right? All you will do in front of instrument, you will just go and put the sample in that instrument. That's all. After that, everything is what you see is what you have to monitor. how good my run is coming, if proteomics is okay, intensity is okay. And at the end of the run, then you will look at the MSMS profile. Right? So even maybe VLC, we have more time, we can run an additional sample with different gradient, or maybe BSA sample something we can run for short gradient 30 minutes. Yeah, ask Avinash or Surbi to show the parameters.
Yeah, I think we can monitor.
So, can you open the, yeah.
This is what they want. It will be, I think, easier if somebody speaks from here rather than they talk from there.
Thank you. Yeah, as just listed, we have to the left side, you see there are two small windows. This indicates the LC parameter and this is the MS parameter, right? So we are right now in the LC module. So there are 5 sub-modules there, starting from sample pickup, gradient, pre-column, and analytical column steps, auto sampler, and easy spray. So is Avinash able to hear us? Can you hear us?
I can.
Yeah. So just ask Avinash to go with the first step, sample pickup and gradient. First, yeah. Right. So here we just get the basic parameters for some amount of that you would want the system to operate at.
I think
It is where you set the parameters for as a right of the solvent along the run, and we go up to as high as around here we say 48% of solvent of solution B, which is 80% acetonitrile in the lab, right? So this comes to somewhere around 30-35% of ACM. And at this hydrophobicity, you see most of the peptides getting eluted out. But the very highly hydrophobic peptides, which are still a part of the column, also need to be eluted out so that they don't interfere with the next run, right?
Sorry,
Which
just a minute. He's sharing in the screen in the on Teams.
yeah. So just after that, you see that the percentage of ACM was ranked up to 80, right? So this shows any leftover peptides which could not get eluted or the highly hydrophobic peptides. Our general experience and the general consensus in the field is that the percentage of these peptides is going to be not very high, which is why they won't really play a major role in your quantification. However, certain samples might have highly hydrophobic peptides, so you might want to take a look at the column that you use and the parameters. Right? So you then maintain the column at a very high ACM percentage and you bring it down so that you equilibrate the column. The success, the major aspect which determines the success of a LC run here is that how well you can condition the column for the next run and how well you can get rid of the peptides from the previous run so that they don't interfere with the next set of samples. We now take a look at the pre-column and the analytical column. So the pre-column is nothing but a guard column. So many times you might have samples. The highest expression of pre-column intake. And then we use a constant flow of 0.5 microliter per minute for the analytical column equilibration. Here, just to give you an idea, the guard column costs somewhere around 30 to 40,000, whereas the analytical column costs around 2 lakhs. So we are talking about a difference of almost 5 crores. So it's okay if you lose a pre-column in the process of saving the analytical column. Yeah. So these are the basic parameters, and now quickly walk you through the MS parameters. So since this is a VDA run, you would first have an MS run followed by an MS, that is the MS2 run. This particular 14. The MS1 will happen in Orbitrap. Then the next round of MS2 would be a result of HCD fragmentation followed by detection again in the Orbitrap. The Orbitrap is a mass analyzer with a detector inbuilt, so it is also it also helps in detection. So to the right side, we see the MS parameters which have been set here. Okay, but before that, you see here there's just one method which has been integrated. So this is MS and MS integrated in one method. Okay. For a DIA based approach or any other PRM based approach where you have MS1 followed by a PRM or a DIA, you will see two such sessions here, right? This, I'm talking about this bar. You'll have an MS1 and then followed by a DIA or a MS2. So here you see the isolation window. So these are all very standard parameters. For someone who is new to this field, you can still get some idea of what each parameter stands for by just hovering over the parameter. You see, Thermo has put in a lot of effort to help the user understand what these parameters mean. And I would strongly suggest, I would recommend that you take a look at these parameters before starting the experiment. Just a minute. Sorry, sir.
You talk about one or two parameters.
Parameters. Okay. Yeah. Right. So in this particular example that we are looking at, the XCD collision energy has been set to 30%. Initially, in the past, so for most of the PD MS experiments, for the LFA experiments, this is usually set to around 30. But for other applications, the parameters will change. So this is basically you telling the instrument or you asking the instrument to fragment at a particular percentage, right? So most of the so if you have a labeled sample like TNT or an iTrack, this number would shoot up to 35 because it needs more energy to fragment the ions as well. Then the Orbitrap resolution. Now there are different resolutions that the instrument can work with. The highest being 5 lakhs. So this is the specialty of fusion, that it can work at very high resolution. This is important for, it's not very important for proteomics experiments. You don't need such high resolution when you do a DDMS run. And usually the resolution is set to very low. The idea here is that higher the resolution, lower will be the data that can be acquired, because the instrument will have to spend that much more time to acquire the data. So if you work at 5 lakhs, you are not likely to see a lot of proteins getting detected. Then the AGC target. So the AGC target also gives the instrument an idea of how many ions to be accumulated before sending them to the detector for fragmentation and followed by detection. So you can, so this is pretty standard, but if you have a certain value to the value in your mind, you can also use the custom option and give the parameter there. Then maximum injection time is the instrument actually takes some time before it sends the ions to the detector, right? So the injection time, so the success of a DDMS experiment, at least in case of an Orbitrap instrument, is largely reliant on the AGC target and the injection time. So you'll have to play with the parameters to understand what works best for your sample and for your instrument. Then if the instrument is very new, then it's pretty straightforward. But as the instrument ages, you have to use slightly lower parameters. Then, I think this is pretty much it. Yeah. So this is pretty much the only last point that I would like to tell you is that the data type. So you have what is called a profile mode and a centroid mode. So in the profile mode, the instrument will take more time to acquire data, but it will be more comprehensive. You will certainly lose out on a lot of other data points that it could have potentially acquired. So we usually operate in a centroided mode for DDMS, and for other experiments which require more data to be acquired, you can go for a profile mode. So this is the basic that I could cover here. And the last step would be just looking at the summary, which will give you all the parameters that were used during this run. So you'll get everything at once. You can use this for your paper as well if you use the instrument.
Great, thank you. It was very nice in a short time. So what we realize that many templates come from these instrument vendors for different type of run. You are doing the data different acquisition or DIA or different type of let's say TMT. So they come with some kind of template, but you have to then tweak around. If version energy was not coming good with 32, and you see MSM is not becoming very good, you can change 35. You can always play with it if this is not very good with the AGC target and all that, right? But again, this will be subject to the instrument and software. So we are don't want to waste too much time on talking every parameter right now. This is a run going on live, right? So that will keep flashing in between so that now we can move on to the progress of it. I think that's it. So after doing all that, what you see is nothing, right? You have no idea what the proteins are. So you have to take the raw files and what Navdeep is going to talk to you about how to analyze the file. Then making software available. We have the commercial software which we use in the lab with the proteome discoverer, PD from thermocompatible. Additionally, the max quant has been pretty popular, but given the time these software take to analyze the data has been humongous. PD takes maybe 2 days time, max quant will take maybe several hours time. So now as a result, the MS Fragger has become very popular because it takes only less than an hour or a few minutes per se. So that's what we'll show.
Okay, so I will start from here. That as Professor Sanjeeva told that it is just a black chromatogram, and we can hardly understand that what is actually there and what are the
See? Windows remote share screen.
Maybe open one window over here, so it will be different. So what we can do is we can shift whenever it is required. Yeah. Okay. So আমাকে শেয়ার করতে দাঁড়িয়ে আসবে। Okay, so we will start the MS Fragger, the frag pipe, this particular software. As I have already told that that we will start from here, and I believe that all of you have reached to this particular interface where all these tools, MS Fragger and Quant and Philosopher has been already downloaded and updated. So why we are doing this is to understand the data that we have just shown. It's a black color chromatogram. Looking into it, we will not understand how many number of proteins are there, how many number of peptides are there. And we have to really make sure that whatever the analysis strategies we are using, we are not losing the actual ion. Rather, we are not taking over the noise. So this particular information is very important for us to understand. So for that, first of all, I request you that you should have the files in one particular folder. We should have at least one control, one treatment, and few of you who have faced issues in the Philosopher, they should have the database which we have uploaded in the drive. Okay, so now I will walk you through each steps. There are few concepts I will tell you, but as we have a pretty less time, so we will do the software we will run the analysis, and we also do this check this particular like the parameter in between. So I will request all of you to come to the workflow, which is just beside the tab of config. So the very good thing about FragPipe is that in the max quant, for the first time, if you are doing max quant, you have to optimize the parameters properly. But TrackPy provides a default parameter of LFQ MDR. So first you can come to this particular drop-down menu. There are multiple already workflows are available based on the applications, which includes even DIA. But right now, as most of you have not much idea in analysis, so we will start with simple LFQ MBR. The LFQ MBR means LFQ communication. And the pandemic match between the runs will not play a big role. But if we are having multiple samples, match between the run plays a major role where it will try to find a peptide in all the runs if a particular peptide in all the samples are not present completely. So this is a very important concept to understand that when a peptide goes into the mass spec, theoretically we believe that all the transitions of the peptide, the data should get recorded, but it actually don't happen in that way. Due to the sample preparation and more technical, due to the analysis, the data analysis also, but it might get only partial record. So during that time, it takes the peptide at least in one of the samples and try to align the peptides in such a way that it comes up with an intensity. So this is the match between run concept. So should I show the run? Okay, fine. So now let's go to this particular part and add the files. So as of now, first we will add the control 1 and treatment 1. So you can add it maybe together. Yeah. And after this, what you can do is that in the experiment, you can give the name. For example, for control, you can give control. And for the treatment, you can give treatment. So for now, I have given just 1 and 2, but it should be control and treatment. Second thing is that the next part is the bioreplicates. The bioreplicates means that if you have multiple replicates of a particular conditions, for example, you have 5 conditions, 2 conditions, each condition 5 samples. Okay. Yeah. So just now we have uploaded the 2 files over here, and the experiments has been set up to 1 and 2. It can even control and treatment. Bioreplicates are very important if you have multiple samples. For example, 3 controls, 3 treatments. Over here, it should be given the numbers based on that. As the data is from the DDA or the shortcut proteomics, it has already taken the data type as DDA. But based on your analysis, you can change it if it is DIA or DDA quant, that means label quantification. Okay, from here we will move to umpire, but we will not do anything because this is for the DIA interface. Okay, if somebody wants to go ahead with the DIA analysis, they can use that. Now the next part is the database. So whoever has, for most of the people, it will show over here download option. For somebody who has not able to download the philosopher properly, so this particular option will not be there. So for them, we have shared the databases in the drive. So you can go to download it, click on browse, and then get the database over there. Whoever is having it, just go to the downloads, click into the human, only use these 3. So the reviewed sequence only. I believe that everybody knows that the UniProt have 2 types of sequence. One is SwissProt, second is Trumbull. SwissProt is the reviewed one, Trumbull is the unreviewed one. We should take the reviewed one forward for the proteomics analysis. Second is the decoys. I will talk about the decoy after this one. Third is the common contaminants. I believe that many of you will be knowing that during the sample preparation, the samples get exposed to multiple, hair, skin, which is keratins, and at the same time we use trypsin, which is a pig trypsin that is also contaminants. Being all these are proteins, and we are living in a world where so many proteins are there in the environment. Lot of contaminants come, and this particular contaminant database has been first developed by Jeremy Cox's lab in who has developed the Maxima. So this particular database generally we use. It's get updated with time. We should not use isoforms. If you want if you're using maybe only want to see some histones or some proteins, then only you select that will need multiple isoforms. IRT sequence or something, if you are spiking in IRT peptides in your samples, then only we should see that. And if you are there will be many manuscripts, for example, the CPTAX manuscripts, where you will see a complete protein has been spiked. For example, a protein from some yeast. So during that time, you have to put the that particular sequence over here. That will help us to do normalization and see run-to-run variation. As of now, we are having nothing in this particular sample. We'll click OK. And the next part is that it will ask that in which directory it will save this particular database to get downloaded. The next part is that the decoyed protein database, which is you can see that it is 40912 entries. However, the total number of proteins, as per we know, in Unicode is 20,000 plus. Then why it has been increased to 40,000? The reason is that the decoy database is something which is non-existent protein database that any data search engines developed in the background. So what it actually does is that it takes a particular FASTA sequence, then it converts into a peptides, or it takes a complete FASTA sequence and it just randomizes, either randomization or reversing that. So once it does that, it is giving a particular sequence which is actually not present in your database. So why we need that? So the thing is that to understand what is correct, you need to understand what is false. So for that particular thing, to understand this your sequence has been correctly identified or not. So it should be there that the false sequence should not be identified more than 1%. This helps us to understand or get the FDR, which is false discovery rate, and it helps us to also get the scores that will give the probability score that I will show in the output file. Once this is done, next we will move to MS Tracker. Most of you are confused regarding whether we are working in PragPipe and MS Tracker. So MS Tracker is the heart of PragPipe. MS Tracker is the search engine that helps to PragPipe to run. Is same as Andromeda is a search engine for the Maxima. Okay. So once you are coming over there is a series of parameters are there. I always love to play with this parameter. I've seen tweaking this parameter really help us to get sometimes good data and to understand even the mass spec instruments are working properly or not just by changing some of the threshold. But at the same time, for like people who are studying new, PragPipe provides a pretty good default parameters which are easy and good to go ahead with. So I will just tell about few of the parameters. The first thing is that the enzymatic cleavage, that is the one which we are using trypsin. Should be a miscleavage should be 2. So what is miscleavage? So miscleavage is something when theoretically we believe that trypsin cleaves into the K and R, but it is theoretically. But actually when we do the experiment, could be multiple things starting from temperature to the efficiency of the particular enzyme to substrate enzyme ratio, that it don't do 100% cleavage in every K and R. So that means it is missing some. And when it is missing some, that particular peptide will not get identified if we don't allow the miscleavage to be 2. Why 2? The gold standard number has been given by HPP, which is Human Protein Project. They have given this number based on looking at the false discovery rate. If it is less than 2, it might lead to a identification of less number of peptide. If it is more than 2, sometimes it helps in the identification of possible. So Next thing is that if it is less than more than 2, the number of peptide sequences will be very small. It again might lead to the false discovery. Apart from this, I will come to the this one, variable modification. So variable modification we generally keep for most of the mass spectrometry data is a modification or like the oxidation of methionine, which generally happen during the sample preparations. But it don't happen in all the proteins. So that's why it should be kept at variable. Second thing is that acetylation of the lysine in the acetylation in the N terminal, which is generally a procedure that happens in our body, which helps in the different chaperones and lifestyles. So this particular thing, there is a paper, very good paper from Ramaswamy in 2018. So that particular paper has talked about why acetylation in protein is important and can help in identifying more peptide in mass spectrometry. Second is the fixed modification. The fixed modification should be carbonomethylation. That happens due to the the alkylation steps that we are doing after reduction, and it should be fixed because we believe that this particular alkylation step should happen in every peptide. If it is not happening, then the reduction step that in the DTT or DTT we are using will become reversible. So for this particular thing, we should use carbonomethylation. Only those peptides we have which has been modified, then only we will take it forward. Okay, next is validation. We will not use this one for the LFQ analysis. PTMs will use that time only when we have either want to look into the PTMs, or second is that we have enriched our peptides. For example, yesterday I think you have listened to many talks about the glyco. Recently, in this particular version, FragPipe has included glyco over here, looking at the lot of work is happening in the glyco. Now coming to the quant MS1. We will select this one because that will help you to identify and get the peptide modification. So we will select this one in the run MS1. All the this particular information will be kept same parameters. Make sure this add max LFQ. I'm keeping it selected, but this actually works when you have a sample more than 3 per condition. Second is that match between the runs should be checked if you really want to see a good amount of data or good number of intensities. MPR, FDR should be 0.01, 1%. Make sure whatever parameter we are using for the analysis should not go less than 1% MPR. Normalized intensity across run should be kept checked, and that will help you to get a data which will give a peptide level intensity which will be normalized. But still, it is good to see that after doing this particular data analysis, whether your data require normalization in the protein level or not. Pre-quant over there, this is an alternate tool. Generally, you can keep it on and off. That's not a big issue, but generally we don't use that in our analysis. Okay, so quant or isometric will be for the TMT or ITRAC. This is today we are not touching on. Spectral library we are not touching on, but it is only based on the validation experiments or the targeted experiments generally we do. Now coming to the quant DIA, this is again same. If you are selecting DIA or umpire in the first step, then only we will do that. But I believe that most of you will be requiring for the LFQ on the level 3 modification. Now coming to the run. So make sure that you are first going to the folder, the master folder that you have over here, and you are making a particular folder which names with maybe output. Yeah, and you are selecting this particular folder because your FragPipe will be requiring a completely empty folder for this particular thing. So if I'm going over there and I'm selecting this, select, and if I'm running the analysis will start running. So as you can see, you should check this particular console. It should not show any errors. If there is any errors we are getting, that means there is some issue we need to understand. Second important point is that you have to be also make sure that whatever steps here it is coming, at least for the first time you should read all the steps. you will really understand that from how these black bushes that just now we saw in the chromatogram is actually getting deconvoluted, and we are getting each of the peptides. According to our experience, it should not take more than 12 to 15 minutes to run the whole analysis. But as we have less time, I will show that how the data actually looks like when we get an output file of it. And we have already shared that particular data for the so that you can take a look into this particular output file, how it actually looks like. Second thing is that we have also shared 3 control and 3 treatment knowingly, so that you can go back to your after the conference and you can see that at how actually and complete experiments analysis look like. Yeah. So I have just dragged this particular output file over here to give you an idea that how it looks like. So this is how our output file looks like. As I told that we have these contaminants. You can see few proteins are coming as contamination. The first one is trypsin, the one which we are using in the sample preparation for converting the proteins into the peptide. Second thing is that we get a list of these IDs, which are protein IDs. You will get all the UniProt IDs name over here, followed by gene name, protein length, or organism you will get based on the database that you will be choosing. Then some description. This is very important, which is protein probability. Max1 gives Andromeda score. This gives protein probability. This is pretty much same as what Max1 gives. More towards 1, it gives you the confidence more. If you are getting some proteins which is less than 0.5 or 0.4, I will say that that particular proteins are not that much important. It's better not to use those proteins. Obviously, the one which will have 0, you have to remove that. After that, you will get multiple information like the number of peptides and all this number of spectral count. But the important thing which you will be needing is the control and treatment. This one. So this will actually help you to get the actual intensity, LFQ intensity, which you will be looking and you will be taking it forward. For example, this. You can ask me why these zeros are there. This is nothing but missing values. So these missing values need to be converted or need to be imputed based on multiple strategies. I think those can be taken sometime else. So this is the result you will get. You have to take these data forward for any kind of proteomics analysis. I have only 1 minute left, but I will try to just touch upon only 2 slides, which will be very important for you to understand that why and how these search engines are working. So the first point is that, so what we are trying to do actually, that is very important to understand. So the first thing is that we are taking the proteins, any samples, tissue, cell line, plant, and we are taking this particular protein and converting it to peptides. So these peptides we are using the trypsin. Followed by, we are injecting these peptides into the mass spectrometry, where we are getting these chromatogram. From here to here, it's very interesting, and we all biologists understand. This is the something which is very difficult to understand. Then how we have to take this thing from this, that is something we are using this particular analysis. So for this particular thing, a particular protein, as you can see, is converted into peptides. Each of the peptides is giving each of the spectra. Okay. So from each of the spectra, when you are using a particular database, it is trying to align the spectra. I think all of you have listened to Dr. Shabdasati's talk yesterday. He told that how a particular sequence can be converted into theoretical spectra. This particular theoretical spectra we get, then it tries to matches the experimental spectra with the theoretical spectra. Based on that, it gives the peptide. It might happen that the identification of the peptide, whatever is looking for each spectra, could be multiple. So for that particular thing, these peptide sequences, one more thing is that this peptide sequence is then taken together and then we get the protein identified. This is how multiple peptides when we are getting, how a particular tool chooses the best one based on the score. That means if you have maybe 8 amino acid and you have got the theoretical spectra for that, how the experimental spectra, how much it is matching. It might be there. The search engine never gives only one peptide. It gives multiple peptide and its scores. But the final analysis results, whatever you got, you will get only that particular peptide which has the maximum score. That means which has the maximum probability score that has been matched, which helps you to get the best match with the score, and all these peptides taken forward to identify a protein. So this is how the whole concept actually works in any search engine you can take. I have given a demo of the FragPy, but if you take MaxQuant, Proteome Discover, all the search engines are built with this particular principle. So I think so that's all from my end. We can take questions, and I believe that you will, many of you will be facing the problems in the analysis, which we can take maybe right now after the tea break. All right, so I hope at least 50% people are able to do it. Those who are not having a laptop right now or could not do that, I'm sure you will try it out. So let's take a tea break right now and continue doing this thing during the tea break as well. So go grab something over there and then continue doing this activity. In 15 minutes time, we will come back and then we have the session to continue. Let's keep an eye on this one which is going on. Let's look good so far. So this is how you monitor in the mass spectrum, right? So almost 1 hour has been done so far. 1 more hour to be done. Okay, we'll take a tea break now. We can play the Miss Wavelength stuff from the things. All right, because everybody's so engrossed, nobody wants to tea break today. So that's really good sign. But please do pick up something if you need to. We will come back.
Ankit, highlight bio-innovation.
All right, welcome back everyone. Post this session. I hope you're all enjoying the Monsoon and the Monsoon Advanced Proteomics School, right? So it's really nice rain outside, and I hope you're now charged with the tea. Okay, so good. Welcome back. So in this workshop, let me just tell you the personal, motivation behind this. So last year I was in California doing my sabbatical at University of California. So during the process, I visited many labs and many places over there. And last month we had a conference, ASMS, American Society of Mass Spectrometry. So my idea was to bring whatever the new, latest, and the most successful things are happening in the North America, bring to India, and at least bring those key concepts and technologies or kits possible in our country, right? So that was the main concept. That's why we thought about bring cell surface proteomics. We are talking to you single cell proteomics and transcriptomics shortly. And different type of reagents and kits which we are trying to display to you, which is all latest happening. So the next 10 minutes, you're going to have a short session from company Bio Innovations. They are going to show you a product known as Proteomics kit. And this Proteomics kit is very user-friendly protein sample preparation kit. What we have been talking to you was a more cost-efficient way of doing proteomics, but very laborious way of doing it. You have to prepare your lysate and each step separately for your, if you want to digest your protein, you have to go through TCEP and alkylation, trypsinization, all of that individually you do. In the process, actually, you lose a lot of sample because your starting material is very high, and at the end, your peptide yield is very, very low. So these kind of kits which are coming forward, they come with the column-based stuff, and you try to bind it and just wash it on the same column subsequently in different steps. So therefore, your yield of the overall protein to peptide becomes very high, and samples are very clean and compatible for the instruments. So I visited few industries over there where some of my previous PhD students are working, and they were using this Proteomics kit. At UCSF also people are using Proteomics kit. Then I thought it's a really good kit. I should start learning about it. And our lab at UCSF also uses this kit. So I thought, let's introduce this in Indian proteomics community as well, and see how this kind of thing can be utilized. So in this way, today Bio Innovation team is going to give a short update on this kit. Actually, they gave a kit to us as well to show in the workshop, but it came very late. So we could not plan any hands-on activity on this. It just came the day before. So, but right now, let them give a glance of the this thing, and if any possibility in future arises, we will try to show you how this kit works. But very user-friendly kit. I have used myself. So, all right, Kamlesh and the team, please go ahead. Yeah. I hope we are audible to you. So we are giving you all the options. Okay, we don't want to bias you to what you have to buy or what you have to purchase or what you have to use. But like S-Trap was one of the options. Likewise, Proteomics is another option. And these are some of the good products which are quite used, right? But in the US market, people only look for the time and success. They don't look for too much cost. Yeah, here you have to weigh out both your cost as well as the success both. Right? But sometime in a mass spectrometry based experiment, let me be very unbiased. In a mass spectrometry experiment, the cost is huge. When you are passing your sample through those columns, which cost 1.5 to 2 lakhs rupees, one column going bad is much more detrimental than buying 10 kits of the same amount, right? So this kind of the trade-off which we have to decide how best we could use it. All right, Kamlesh, I hope we are audible to you. Please go ahead and start.
Yeah, thank you very much, Professor.
You're not audible as of now.
Yeah, for giving an opportunity. So let's start that. Okay. So is my screen visible?
Kamlesh, we can't hear you.
Yeah. Hello.
Please unmute or is there any other system we can use? Which can we hear him in any other computer?
I can hear you, sir. Can you hear me? Hello. I can hear you, sir. You can hear me? Hello? Hello?
Kamlesh, can you raise your hand that you can hear us?
Disturbance mode.
Yeah, I can hear you, and yes. Hello.
There.
He is audible in Teams, but not in.
Oh, he's audible in Teams. So
Hello.
Kamlesh, for 5 minutes, we are going to figure this out.
Okay. Yeah.
Is my Kamlesh?
Okay.
Looks like you're audible to the online audience, but not the offline audience here.
Okay.
Okay. Please speak again. Looks like it has worked now. Speak again. Looks like it has worked now. Kamlesh, please start.
Yeah. So thank you very much, Professor Sajiva, for giving an opportunity to discuss about the latest solution for sample processing in in-depth proteomic analysis. And let's start. So Proteomics is actually a spin-off company started in Germany and from the Matthias Mann's lab. Okay, and Dr. Gerwin is a co-founder of it. Okay, so he's you know that the man behind this company, okay, the Gerwin and Dr. Matthias Mann. Okay, so what Proteomics portfolio is mainly like to simplify your sample preparation workflow. Okay, and for your entire workflow of proteomics, whether it is like academic or CRO or biopharma. So like we all know that like nowadays world is moving toward tissue as well as specialomics for biomarker discoveries. Okay, so we'll start with the simplest workflow where we have a bit box which is like here. Okay, so this bit box is like just let me start my introduction. Okay. So this bitbox is a homogenizer which has been used for in a workflow for your proteomic sample preparation. Okay, so you just homogenize, extract, and then you use IST kits, which is kits which is mainly been used globally for proteomics sample preparation. Okay, so these are the three main areas where people are using proteomics lab, like R&D research institutes. Okay, then characterization, biopharma, and then CROs for PKPD and DMPK. So I'll quickly go that like this is what exactly we are doing currently, like in-house workflow where we lyse the tissue cell or we then digest and then further purify. Okay, so these are the workflow for biopharma for protein characterization, lyse, digest, and purification. Okay, so what Proteomics has? Proteomics has the homogenization system, that is a bit box for those who are working on tissue biomarker or tissue proteomics. Then we have a kits, that is IST kits. Those who are working on blood or liquid or any other cell lysate. Okay, then we also have an automation. Okay, which is a fully automated workflow, which can be also aligned with your existing system from Agilent, Hamilton, or TCAN. Okay, and then the latest is the kit which we have launched in ASMS 2023. That is like enrich plasma serum proteomics related workflow where you can enrich as well as you can purify the protein. Okay, so this is like four different product line we have. Okay, so let's start with the bitbox. So bitbox is like from 24 to 96 samples. Okay, it is homogenizer. So unlike your sonicator, it will give you more yield in a protein. Okay, which we can. So we are not going more on a specification side. Okay, so let's what exactly bitbox can do. And bitbox is a small homogenizer kind of system, but specifically designed for the proteomic studies. Okay, it's not like a routine homogenizer what we are using in the lab. Okay, and it can be combined with the bitbox. Bitbox can be combined with the IST kits, and it gives very reliable result, and the protein IDs also increases by using bitbox, especially on FFP tissues. So this is a workflow we can use like for proteomic studies, especially for like those who are working on tissues. Okay, so any kind of tissues, FFP, fresh frozen, okay, and it can goes to homogenizer, tissue homogenizer, okay, which we say it bitbox, then the protein will be extracted. Then we follow IST digestion cleanup protocol. So IST digestion and cleanup protocol are pretty optimized, and it has been used since last 8 to 10 years. Major US and Europe institutes as well as pharma companies, they are using this. And then finally that purified protein, okay, or the cleanup peptides will goes to MS. So the beauty of this particular system that it require a very minimal sample when especially you are working on needle biopsy or that some of the FFP tissue. Okay, so this is how people are using it. Okay, this is like some of the comparison study which we have shown, like where that normal sonicator and like bitbox, how exactly it will make a difference. So you can see that the bitbox, using a bitbox, the sample preparation, it will increase the like overall protein ID, okay, as well as the protein yield also. So it is pretty simple workflow. It will reduce the time from 9 to 10 hours to 10 minutes, okay, and the total time for sample preparation is reduced to 5 hours. Okay, so it will work, as I said, that like both on deparaffinization as well as on cull FFP tissues. Okay, so this is what the workflow is. So xylene free as well as like deparaffinization has been already done. Okay, and this is a comparison which we have shown that normal sonication, how much time it will take, as well as what is the time it will taken by bitbox. So like we have tried to show that like different tissues from mouse, rat, as well as from human, okay, and spleen, kidney, liver, muscles, okay. So you can see that like this is a xylene based deparaffinization workflow where we have the protein IDs has been increased, okay, as well as a full curve where like you take a section of FFP tissue directly and you use that without deparaffinization. So in both the cases, it will increase the protein IDs. Okay. So this is a simplified workflow again, and then it shows the mouse liver related samples which we have processed, and then it shows the increase in the IDs. So it is a comparison between like two different workflow of direct curve FFP tissue as well as that defertilized. Okay, so it shows very reliable protein intensity, okay, as well as the IDs. High quality in depth, wherever you have a discovery mode analysis for the proteomics as well as peptidomics, then you can utilize this system, especially for the cells or difficult tissues. Okay, so this is what. Now we will move toward the IST technologies, which is a core technologies. Okay, in case you do not want to use bitbox, or if you have a soft tissues, then straight away you can use the IST kits, okay, which do not require any specific instrument. So what exactly it will help and how exactly it will help? So in a common workflow or lab developed test, you must be knowing that like the total time, okay, maybe like more than 30 hours or 40 hours. Okay, so especially precipitation and trypsin digestion took a major time. So Proteomics has developed innovative technology using proprietary reagents where we reduce the time from 44 hours to 2.5 hours. Even the hands-on time, okay, from 4 hours to 1 hour. So this is like something which now people are interested to reduce the time, as well as without disturbing their workflow or without disturbing their overall result. Okay, so this IST technology kits, which been used in a laboratory, do not require any specific instrument. Your lab equipment like centrifuge can be used, and you can achieve that particular results. So another is like preomics, okay, automation. So we have a Prion machine which is fully automated system. In case you have a large amount of samples, okay, and where you don't want to do hands-on, like you don't want to waste your time on doing like hands-on time, then this machine can fully automate your workflow, and it will further reduce your hands-on time. Okay, so this is Prion system. So we have a different type of sample matrix which has been used using IST. Okay, so it can be used for tissue, it can be used for your biological fluid or plant or whatever samples you have. Okay, so different samples has been used, okay, or can be optimized, okay, using this particular kit, and it gives desirable result using any mass spectrometry for the proteomics. Okay, so these are the three main fast moving items what people are buying globally. Okay, protein sample preparation as well as biological fluid. Okay, so those who are working on plasma proteomics or serum proteomics, okay, or those who are working on protein interaction, that they also use this kind of kit. Okay, so this is some of the result what has been published using IST kits or IST technology. Okay, it clearly shows that bitbox or as well as the control as well as normal kits. Okay, so using Prion also we have identified some of the samples. So human plasma, you can see that it can increase the protein ID. So this is one of the application not we have published using IST kit as well as Bruker's timsTOF machine, where it shows very good concordance, and especially for the IAPSF workflow, okay, where the protein IDs has been increased as compared to normal workflow using IST kits. Okay, so this is we can share that particular app note for you, and so especially in IAPSF where the short gradient, okay, so short gradient as well as that IST technology has helped them to get the desired result.
Kamlesh,
Yeah.
So thanks for the overview. I think, in the interest of time, we have to stop here.
Yes. Yes.
The main idea what to be conveyed, I have already conveyed to the audience, and we have seen the data here as well.
Yes.
Yeah. So sorry to interrupt you now, but I think, we are running a bit late on the session. So if it is okay, we can stop this here. But, the key message is clear that, it is very user-friendly kit, and a lot of laborious steps can be reduced here with the, as you mentioned, 2.5 hours. And our efficiency of doing the steps are very high. So those points are well taken. So thank you very much for bringing this kit in India. And now I think users can probably support it.
Thanks, sir. Okay. Thank you very much.
Thank you. All right, so we are going to talk to you very briefly about single cell proteomics. Again, we have never done it. It is as much I'm new to this field as you are. But what you are seeing, probably the way field is progressing, there is more and more emphasis about looking at single cell and also improve the coverage from the single cell, right? How best one could try to get the maximum coverage from the given single cell. And if we just think about how the proteomics is actually evolving, I'm sure biologically you will appreciate that each one of these cells has their own uniqueness, and one would try like to get the proteins from those single cells. But our ability of doing the proteomics was not as high that we can get a lot of protein from the single cell. It was hardly few hundred proteins till the Bruker's Tim-Stroff instrument came, and they brought a model, SCP model of Tim-Stroff. So now our, very quickly after, this time, if you see like 2020 onwards, it has really, reached exponentially now. And we started talking thousands of a scale of single cell, you can identify 1,000 protein. Now they have really pushed it high, made it to 3,000. And for the first time I saw Astral data, they showed 5,000. If you from single cell, you are now able to get that kind of number of protein, right? Which is still very difficult to get from the global proteomics as well. But nevertheless, I think we have to now see how best we could reach out to the biology by looking at the very actual cell from where we want to understand any problems. So there are many challenges for doing single cell proteomics. We have to think about sample preparation. Here the single cell we are talking, so sample volume is very low. Your initial quantity of sample is very low. And that's a major challenge which you will face all the time. How much protein, how much peptide in reality, right? So how much starting material you have to go? 10 million cells, 50 million cells, how many cells you need? So that is one of the challenge. And there are many innovative methods have come forward to improve the sample preparation strategies. Chromatography, you always need to be optimized. Shorter gradient, maximum coverage you want. Mass spectrometry, how best to, now the number of proteins will be less, but still the abundance is very low. You want to improve that coverage. And data analysis, many of the strategies coming forward. So these are many challenges in this area. Sample prep, as I said, is one of the major challenge. And there have been many innovative studies and the methods came forward, which have tried to address this problem of nanopots is one of the very crucial studies which I will talk in subsequent slides. The idea is to use a nanoliter dispensing system to reduce the volume and less sample can be taken forward. How best one could take the single cell and try to do the quantitative analysis from those. So you have heard about TMT, tandem mass tag yesterday, about one of the ways of doing quantitative proteomics. Some people have utilized these tandem mass tags for looking, how best we can utilize for the chemical labeling of single cell. There's a company which has come with this product for Selenium. Again, that is a good way of manual sample handling procedure. And then, of course, it was all not possible to talk single cell proteomics. From this year onwards, you will see a huge number of studies and work coming out because of these instruments. Because earlier, whatever sample prep you do, you are going to get few hundred proteins. So you can't talk about proteomics or proteome wide scale. But now, given these major mass spec instruments have come forward, now you will see very soon, when you talk about biology, people will ask you, can you do single cell proteomics? Your reviewers are going to very strict in that regard. So why that becomes so important to look at single cell for a specific biological question? A nice review article from Tammy Giger. Here you think about, we all want to look into different cancer biomarker or different disease biomarker per se, right? But now if you come and look at how heterogeneous this mixture is, that solid tumor mass, and it contains various types of cells. So while you are only looking at the proteins coming from the cancer cell, but along with that, you are looking at T cell, macrophage, neutrophils, fibroblasts, blood vessel, many things together, right? So then eventually what proteome you are analyzing, cancer cell is only one subset of that, of the overall proteome, what you are obtaining from this. So if you can segregate these cells and only enrich the cancer cells and then try to extract the protein from the cancer cell, then I think your biology is going to be very specific and not going to be confounded with the other cells, right? So that's where this kind of area is coming forward. Conventionally, what approaches have been used, antibody-based approaches, how to couple your site of mass spectrometry, especially induced couple plasma mass spectrometry, use that for analyzing this kind of single cell. Single cell Western blots or some of the other reagents, which is a PCR-based immunoassay kit, all of those were part of this kind of single cell analysis. A site of mass cytometry is one of the popular methods being used. When you are using the flow cytometry based approaches, you are labeling your different antibodies with different heavy metals. And in the process of analyzing your ions, you are looking at those heavy metal ions with the TOF. And therefore, you are able to look for, by enriching your samples with the different type of flow cytometry method followed by mass spectrometry analysis, you can identify single cell. But this is again an antibody-based approach, right? You are looking at enrichment with antibodies. There is a very big resource available for looking at single cell human protein atlas. But again, that is a resource which you can't replicate in your own labs, given that it requires a lot of antibodies. So again, if you have good antibodies which can enrich the single cell, but this is a good reference point for you. It is open access resource available for you to look at different biological questions. But the entire field of single cell proteomics really got evolved with Scopes method, which came from Nikolai Sevok labs. And the first time they showed how best you can utilize the existing tools available, but just make some fine tune in different things in sample preparation strategies. Looking at, they try to use the sonication system from Covariance instrument model to harsh lysis and sonication in the glass, and then they use the capillary electrophoresis methods rather than the standard LC columns. In the Orbitrap or the mass spec part, they again played many small changes to improve the overall ion accumulation, because from the single cell it was less ions are coming. So they played with those, and they introduced a new concept of the carrier cell, which I'll talk. So in this way, they were first time able to show with the Scopes method that now they can analyze a single cell proteome. So again, in the Orbitrap, also like they were trying to play with the ion accumulation parameters. Typically, we use 20 to 50 milliseconds, but now they said, okay, number of peptides are less, but we need more ion to be accumulated because the number of peptides are less from each cell. So how to improve that? They spend more time of the instrument to enrich those peptides. So therefore, coverage, they were not too much worried. It will be low, but at least the peptide confidence level should be high. So they played with these parameters. And then they use the TMT tags. Idea was, can we label the single cell with the TMT tag? And if we are comparing different conditions, in this case, I think they took two different cell lines. This one of the, I'll show you the cell lines. These were U937 and Jurkat cells. So these two cell lines they chose, and idea was to look for their quantitative analysis. So the concept was, can we take the single cell, label them with different reporter ions of TMT, which are isobaric tags, and then try to do quantitative analysis from this labeling, right? After enriching a single cell, label those and do the MS and MS analysis. So one cell, one cell type and another cell type. So these two then, what they use only 8 single cells. So again, isolating single cell will be challenging. So how many cells we need really for doing this kind of proteomics? They tried minimum 8 single cells, but again, this is not going to give you sufficient coverage for doing mass spec and proteomics. So therefore, they made this carrier cell, which is 200 cell. Now they have combined from both the conditions. And this is also they're going to run. This is also going to be labeled and run. Now this is going to improve overall your peptide and the coverage for doing the MS identification part, right? And these will be used for your mainly quantification part. So this was the experimental design. They used U937 and Jurkat cells. One of the lane or the one reaction, they kept it for the empty cell, and 131 channel they use for the carrier cells. It means the mixture of 100 cells of Jurkat, 100 cells of U937. So in this way, the experimental design was good, and now they are doing quantitative analysis. All these are different reporter ions of TMT, tandem mass tags, which are all isobaric, and you can now use to see the unique pattern from these cells. So briefly, the result what they found was they were able to segregate both of the cell types, right? They have isolated single cells, and now using quantitative approach, they're able to show that they can detect in the mass spec and they can segregate these population very well. Additionally, they also showed they can do quantification, like they are able to quantify for every protein, like different protein, what happens to their signal in both the cell lines or cell types. So now it was very milestone paper in the field, which showed that okay, with the existing method you can do the sample prep, play with the MS parameter and chromatography methods, and enrich these and label with the TMT tags and do the single cell proteomics. But there were, of course, many small challenges involved. So they came up with the another version now, which is known as the scope 2 method. Now they again now further worked on the sample processing steps, try to improve now the freeze thaw heating cycle. And then they have also used, of course, FACS to sort these cells. The reference channel or the carrier cells were improved to 5 times to improve more coverage. And earlier, the other issue what they have was they ran sample 3 hours gradient to get the coverage. Now it was run in 1 hour gradient. So again, many of these things were changed. And idea was that they can still do single cell proteomics with the shorter gradient and good coverage can be obtained. One method which came very popular in the field is the nanopot. This was a Nature Communication paper. How to prepare your samples for single cell with the very little volume available, right? And here you don't want to lose the sample. Again, of course, in any proteomic sample, you don't want to lose the samples. You will always be having the precious samples from mostly a lot of clinical samples or even drug-treated samples. But in this cases, you are talking about, few cells, 8 cell, 10 cell, or, So in this case, from the same, this was the NanoVel pattern slide they have prepared. And on each of these nano droplet, this entire reaction has to happen. So it means you're not going to lose from transferring one tube to other tube and then going through the different steps. So, right. So sample evaluation, looking at the facts, looking at now the reduction step, alkylation, digestion with the 2 different enzymes, cleavage and peptide creation. Everything is done on the same spot. So this is nanopot, and that became quite popular. Now there are commercial chips available in this method. Now the field is still evolving, and idea is how to improve the coverage with the newer latest mass spec methods. So what was earlier data dependent acquisition or what was used in the Scopes paper was TMT method and the DDA method. Now people are trying with the DIA method. So this is the work coming from Matthias Munn's lab. It's still unpublished here, but they have referred this in this review article of Nature Methods. If you are keen on this, which I think you should start reading about it, this year of March issue, there is a full special issue Nature Methods on single cell proteomics. So think about, recognizing the importance of the evolving field. Editors have came up with a full special issue of Nature Methods, if of this year March, when they have now featured many articles, and Matthias won the first perspective article that talks about different methods. So they are working very actively on it. This work is still unpublished. But what this means, they are not using TMT tag. They are using non-isobaric tagging strategy. And again, the cells are enriched for, let's say, 2 different cell populations are enriched, and a reference pool is made. So the concept remains still pretty similar to the original scope method, but the labeling strategies have been changed, right? Now they are labeling with the dimethyl labeling, which is going to show the 4 Dalton shift. So that is one major change here. But second is they are not using the DDA method, they are using DIA method. And since they are using now the dimethyl labeling, they have termed as a MDIA. So this is the new advancement happening, and Matthias Munn is always known for pushing the boundaries of proteomics field, right? The coverage goes beyond what any lab start thinking about it, right? So now they have first time I think commented that they can reach up to 5,000 proteins with this method of MDIA and using this kind of labeling. So good idea for you to read about it, start thinking along these lines. If you are somebody who's starting your career in proteomics now, why not start with the best? Why you have to go back to to the 10 years old technologies and then take that path, right? You start with the DIA, start with single cell proteomics, and then learn in that area. It is very much evolving, and now I think you will have a good career in that area. Likewise, an additional method again from the Nikolai's group, but they have been much faster or the more smarter in this case than other groups. They have already published in Nature Biotech this year. So they have come up with a new method, Plex DIA. Same concept what Matthias Munn other team is working on, but they have termed the Plex DIA. Again, you are enriching, let's say, cell populations. You are labeling with 3 different dimethyls, and now you are mixing them together to make a reference pool. And then further, what they showed that you can make in the DIA, you have to first make the DDA library. And so they have run the 3 samples separately to build the library, and followed by now they can do the Plex DIA. It's a different name, but almost everything remains same, right? So again, this concept is now going to catch up. To use DIA, multiplex labeling, and then do the single cell up to 5,000 protein is what going to change the proteomics field. MX imaging, of course, we don't work in much in this area, but it's all independent field itself to look into how imaging can be utilized to look at the single cells. And there are many methods, like I was amazed when I was trying to dig deeper into this area, that there are many methods available to look at single cell with its different type of mass spec based approaches. One is of course imaging mass cytometry approaches. Then we have multiplex ion beam method. We have the SIMS method or secondary ion mass spec. MALDI is already established from long time. Different versions of MALDI is available and ESI is available. So there are so many parallel methods of looking at imaging based thing to look for single cell. Of course, again, Maxius1 team, they push the things to a different level. They're also now thinking how to take FFP kind of samples. And every time you keep hearing FFP is one of the most challenging sample, right? Everybody who want to show their innovation, they say, okay, I can do FFP. So how to take FFP kind of samples, where you have the tumor lump or tumor mass there. And now from there, you have to do the single cell extraction, which is very challenging, right? So now they are saying that if you can use some markers for that cancer cell or which you are analyzing or want to analyze, using the deep visual proteome analysis, a software which they have created, DVP. From that, let's say you are having this, FFP tissue block, and now from there, using the marker which can stain the single cell, and then coming with the deep learning approaches, you then focus on those regions, and then only excise those cells from there. And now you can do the single cell proteomics. So just I think just be open and read all of these new innovations. Again, it's probably unpublished paper. It is part of the other paper which is in the right now in the open available. So there are many advancements happening, and not only single cell proteomics per se, but also how to analyze the whole proteome, right, with a single cell molecule. And right now what we have been doing, everything has been dependent upon mass spec to look at peptide sequences, right? There's a set protocol, and now there are too many vendors available to do the similar things, just by tweaking around very in a different way. But the mass spec costs have been very much difficult for the small labs to adopt that, right? So therefore, now there are newer approaches coming, protein sequencing technologies, which is like at least 4 of these are the major player right now: Glyphic Biotechnology, Nautilus Biotechnology, Oxford Nanopore, and Quantum Sci. While these are all upcoming technologies, but one has already reached very close to market. We have met these guys at UPO conference, and I wrote this number because I asked them how much it costs, $70,000. Not too bad. If you think about, every lab in India can buy this instrument, right? So not that I'm saying you should buy it and it is the best instrument, I have not used it, but at least the think about how the field is progressing rapidly. Now you can do at least, for large number of protein sequencing with very, short cost or the less cost. And this is actually based on the semiconductor protein sequencing based method, which is a very interesting article, a lot of interesting thing to talk in that, but I have cut short my talk time. So I'm not going to that detail, but I can refer you to the science article where you can go through it and you can see how best they're doing sequencing. So a science article by Jonathan Rothberg and people are available, but the instrument is already available. That is actually good. So field is pushing very hard. Other thing which is not in the market yet, but will be upcoming soon, Nautilus Biotechnology in the Bay Area, California. They are working on one of the chip-based platform known as the PRISM, or the protein identification by short epitope mapping, where they have made different type of multi-affinity reagents, which can label with different amino acids, and therefore using their deep learning suite followed by experiments, they can actually come up with the whole sequencing. And this will be probably first non-mass spec based method, which can aim to look at the entire proteome sequencing. So again, things are really rapidly fast in the field of proteomics. You have to keep up the pace, right? You can't just be sitting into the very old workflow then and say I'm doing great. So the last slide, just to summarize. Again, I have not done single cell myself, but will be very soon starting doing it, because now I think there is a good motivation. There is a lot of good workflows available, and one should push themselves if you are thinking about disease biology per se. This is the field to move forward. So sample preparation, it's not very difficult. I think those are standard sonication lysis method which you should use. Of course, problem will be how to ensure your single cell is single cell and how many cells you have, and starting material. Those things you have to do a lot of QC checks. And then sample handling, sample prep volume will be challenged. But given that now the instruments are getting more sensitive, methods are available, probably it's a good idea to move forward in this area, right? So that's all for this presentation, to give you a glimpse of what is happening in single cell proteomics. But what we have really evolved well in the single cell area, single cell transcriptomics. Okay, given that the transcripts are representing good functional information, So single cell transcriptomic field has evolved quite robustly over the decade now, and single cell proteomics just started now. So what we thought, while the focus of the workshop is on the proteomics, but we still wanted to give you some flavor of other omic technology as well. So in this slide, we thought to continue the single cell field and bring now the single cell transcriptomics. And that's where I have invited now Dr. Saket Chaudhary. So I'll end my presentation here and let him continue on the single cell transcriptomics. Thank you very much, everyone.
Thank you, sir, for the really nice talk on the advancing single cell proteomics. So I would like to take this.
Am I audible now?
Yes.
Okay, I'm at currently a postdoc at the New York Genome Center where I work on developing methods for analyzing single cell data, and we'll go over the workshop over some of the methods that we have developed and how we use that to analyze transcriptomics data. Before we get started into the presentation, just a very shortest introduction that you need to get into this field is the idea of what happens as part of what Francis Crick coined as a central dogma of biology, that once information flows from DNA to RNA to protein, there's no way it can flow back. And the focus of the talk today here will be how do we measure the information that is there in RNA. So the quick focus here would be how the DNA gets transcribed to RNA, and the RNA essentially ends up making protein. Before we do that, I'll just clarify what transcriptome actually is. So the total RNA of the cell can be divided into two broad categories, one being the coding RNA and one being the functional RNA. The coding RNA just being 4% of total. And that is what we'll focus on. There is also the rest, 96%, which includes your tRNAs, your RNAs, your siRNAs, your miRNAs, which will not be the focus of technologies that I'm going to talk about today. Doesn't mean that they are not important. It's just that the focus right now is on the coding part. Very quickly, what happens during the process of transcription is you have a single-stranded DNA, which gets transcribed into RNA that has introns and exons. This happens in the nucleus. While it is happening, the transcript will also undergo modification. You'll add a cap, you'll add a poly A tail, which is towards that bottom. And that is also the key of a lot of technologies that we're going to talk about today. How that poly A tail is utilized to capture the just the coding part of the RNA. And then this gets under the splicing machinery, which makes your final mature RNA, the mRNA, which essentially gets translated into protein. So a central goal of a lot of biology that we do is we want to understand what shapes a particular cell type, or what is the molecular diversity doing, and how it defines a particular cell type, and how it ends up making a particular tissue. What I'm showing here is a brain which is made up of hundreds, if not thousands, of different cell types. And our goal is to understand what are these cell types really doing. And this has been made possible in the past couple of decades. Like, this question has been there in the heads of biologists and computational biologists for a while now. But it was possible to measure, it became possible to measure RNA at scale only in the past couple of decades. And this started from microarrays. So no introduction to single cell or RNA-seq is complete without talking about microarrays, which were a low throughput method of essentially measuring what transcripts are being produced in a particular sample. And the sample protocol will not go into the detail, but briefly, what microarrays have is you have a slide chip, you have probes that are pre-decided based on the sequence hybridization. And once you have your sample, you will the transcripts in that sample will get hybridized to this particular chip, will emit a signal that can be measured. The challenge, however, is you need to predetermine what those sequences that are sitting on your glass slide should be, which makes it low throughput, and it limits the amount of signal that you can essentially measure. So back in 2009, development of a technology which is called RNA-seq or mRNA-seq, which stands for RNA sequencing, changed this paradigm. The idea behind RNA sequencing is to leverage developments that have happened in next generation sequencing to measure the transcriptome of the cell. Briefly, the protocol is very simple. You have a sample which has RNA. You will particularly select for those transcripts which have a poly A tail, which again is true for majority of the mRNAs that we have. Once these poly A tail fragments are selected, you can convert them to cDNA, fragmented. These short fragments can then be put onto a sequencer. You end up sequencing those. And once you get these short reads out of that sequencer, you can go back to your genome and find out where exactly do they align. And that tells you for each gene, how much amount of signal are you getting. One thing to keep in mind for these technologies is these technologies are all okay. Coming back to what is. Sorry. Okay. What is possible using this technology is to get a view about what are cis-equilials, for example, which is what particular mutation in particular individual gives rise to change in expression in the associated gene, which is all great. But what has happened in the process of developing this technology is we have lost sight of individual cell types. That is not the resolution at which this technology was originally designed for. So what I mean by that is, imagine you have this complex tissue, which is brain, made up of different Lego pieces, which are, in this case, representative of different cell types. When you do a bulk RNA-seq or RNA-seq, which I talked about, I'm referring to this as bulk because it's done in tissue. What you get out of this sequencer is an average version of the molecular profile that the brain has, which is great. But there are challenges that this particular technology pros is if your questions that you're asking are mechanisms that act at a cell level. So if I'm interested in, for example, understanding how gene A's expression changes with respect to gene B, if I were to do a bulk RNA-seq, I'll not get the identity of the individual cells that I'm measuring. So I'll end up measuring essentially that this gene A and gene B are negatively correlated in this case. However, if I had access to a technology that would also tell me what is the individual cell type of this particular cells that I'm measuring, then the correlation pattern can totally. Tissue, but at the same time retaining the molecular heterogeneity of individual cell types. So you still get back your Lego pieces, but now your Lego pieces are arranged by colors. And these colors are indicative of the cell type that that particular Lego piece represents. So if we were to go back to our thought example of measuring individual genes expression in individual cells, imagine you have these 3 different genes, which have 6 different, which are expressed differently in these 6 different cells. If you were to perform a bulk RNA-seq, all you would get is 6 molecular counts for each of these genes, which would mask out the variation that actually we can see clearly in this figure. And that variation, one of the variation patterns that you can clearly make out just by using your eye is the blue and the red never co-occur together. But if you do a bulk sequencing, this pattern gets masked out. All you see is everything happens to be together with everything. If we do a single cell, that covariation pattern that exists between 2 different genes is something that's more repeated. So that is the power of this technology. So single cell RNA-seq, the first sequencing of a single cell in this high throughput fashion happened back in 2009. So RNA-seq was developed close to 2008, towards the end of 2008, and just 1 year later, there was a paper which did exactly the same protocol with slight modifications. But rather than sequencing one entire tissue, just sequenced one single blastomere. So blastomeres are slightly easy to handle because they're bigger in size. So this protocol was not optimized for single cells. So the goal of this workshop is to introduce you to some of the technologies that have been developed in the past decade that allow this scale go from literally one cell to hundreds of cells to thousands of cells in just one experiment. So the first paper for single cell, which I showed just in the last slide, came around in 2009. In the past decade, this technology, as Professor Sanjeeva also talked about, has actually scaled literally exponentially. There have been a number of technologies that have been developed all the way till this is not an updated plot. This is from 2019, but there have been a number of developments that have happened that are still happening in the past couple of years. To give you a glimpse of what I mean by the scale at which things are moving, we have trillions of cells are in our human body. So thought experiment is, and something that you will probably all relate to measure scale is something called Moore's law. Moore's law states that every 2 years, your number of transistors that you measure on a single chip, your Pentium chip, your AMD, or so on, will double up. So if we plot the number of transistors that have come out in the past 2 decades, you can clearly see there's a linear scale here, but the y-axis is log scale. So this has been exponential. Okay. Now this is the slope of how the number of transistors have been growing. Let's look at what has single cell profiled, right? So each dot here is one single cell study coming from which is published. And the number here indicates the total number of cells that particular study profiled. You can clearly see the slope actually beats Moore's law. So it's scaling at a much higher rate as compared to what would be predicted even by Moore's law. The total number of, this is just the total number of cells being profiled. There are right now 2,000 plus single cell RNA-seq datasets that have been deposited in public that are free to use. One thing that happens in this field pretty quickly is, since you have so much complexity in the data, which we'll go over during the part of our workshop, there is a requirement of a number of tools that you need for analyzing this kind of data. So while the number of data sets have scaled, which is what is shown on the x-axis, the corresponding number of tools that you need to develop to analyze this data has actually also been scaling almost linearly, which is interesting. So what has happened in the last 2 years is there has been a lot of effort from the community to organize these data sets. So what was happening almost 5 to 6 years back in this field was everybody would do a study, would put out their data somewhere on public, but it was more or less unusable because for analyzing any kind of this data, you also require metadata information. In the past 2 to 3 years, there have been a number of efforts from different people, from some consortia or some private companies, one of them being CZI, which has come up with portals for analyzing, for putting out these analyzed or partially analyzed data sets, which you can download. So just to give you a brief view of how I work now, if I have a hypothesis, I go to one of these websites, I download the dataset, which is what you will do today as part of your workshop as well, and just go through the hypothesis that you have in mind. So it's pretty easy, pretty quick to check whether something that you have in mind, does that hold in a single cell setting or not. So what has single cell enabled? So single cell, if we were to go back to our brain example, it gives us a view or a measure of individual molecular profile of individual cells. So the brain is made up of different cell types broadly, which can be classified into neurons, non-neurons. And these, if you have this thing, if you can perform a single cell RNA-seq, you can get individual profiles of neurons, oligodendrocytes, microglia, and astrocytes. This microglia cell type will become more relevant as we move along the workshop. What this allows us to do is it allows us to measure, in particularly, if you have a disease or a case to control setting, which particular cell type is the one where the gene expression is changing. So if you have an Alzheimer's disease brain, is it the entire brain that's changing, or is it just one particular cell type where you see most of the gene? That is the resolution that this particular technology allows us to measure and allows us to figure out which particular cell type is undergoing change. One of the key applications of this technology, of course, given that you have the ability to measure single cells, is to be able to find out rare cell types. So if something, right now what I'm showing you here is something that has been known to neuroscientists for a while. But inside neurons, are there specific rare cell types that have appeared in a diseased person that this technology allows you to figure out? There are also a bunch of other applications, which unfortunately we'll not have time to talk about in the workshop. But one key application of this technology is to figure out how one particular cell type makes other cell type. Or if you have a progenitor cell population, how does it make progress in terms of finding its final fate? This broadly is also the category of trajectory inference, which we'll not have time to cover in the workshop, but I'm always happy to talk about how the methods work. And one key application, which is a wide scale application and is something that's being worked on actively, is can we now sequence individuals, figure out what their cell type is. At the same time, if we were able to genotype them in some sense, can we figure out how the genotype relates to expression in individual cells? So does the gene expression change based on what genotype you have, given that now you have the ability to measure the expression of genes in individual cells and individual cell types. So given we have this way of measuring single cell technology, what does an ideal single cell technology should look like? So the first thing that it should have is it should have a capture rate of 100%. It should that every input cell that you give it, it's able to sequence it and give it out the profile of that individual cell. It should have 100% sensitivity, which means that if you were to sequence the transcriptome of that particular cell, every transcript that is there in the cell gets sequenced. It should have very high specificity. So if I mean by that is it is the reason being the technology is still noisy. Just measuring one single cell tells you very limited about what is happening in that cell. If you cannot sort of get an idea of the variance at which those observations were made. So what happens in this field is you measure, the field is called single cell technology and single cell RNA-seq, but the inferences are made on a bunch of cells, which have a very similar cell state. Again, will become clear as we move along the workshop. It's also not entirely, so single cell is also not entire cell, which means that not all molecules that are there in the cell gets captured. There is always loss that's going to happen just because there is loss that happens in any technology. So it's not the entire cell that gets measured. And finally, it's even not RNA. It's not RNA in the sense that what we end up profiling is actually the cDNA, which is a good proxy for RNA, but there are losses that happen there as well. And again, it is not an absolute measurement. So everything is relative. You cannot say if you get 10 copies of a particular cDNA molecule in one cell that it expresses 10 copies of that particular RNA. Okay, so with that figured out, we can now get into what are these different technologies. So this list is ever growing. It's a non-exhaustive list of technologies here. There are multiple columns. We'll focus on 5 of these during the course of presentation. 3 of them are very related. 2 of them are slightly different. What I want to, however, focus on is the scale that you see here, which is the number of cell types that you can measure using these technologies. So the world of single cell started around by measuring 1 cell and then went on to measure 96 cells at once. And at this point, it has reached almost a bar of 10 raised to 5 in just one single cell experiment. And you can always do multiplexing, which can put this bar close to the scale that I talked about earlier. So broadly, the technology, the single cell technology begins by separating, you have these cells that you want to measure in their individual state. So any protocol that I talk about has these key steps. The first step is you want to physically separate the cells. Here I'm physically separating them into wells, into plates, which is one way to do it, but this is just representative. So all you want at this point is different sets of each individual cell should be physically separate because we want to carry out reactions in these chambers. This reaction will happen once you lyse the cell. So you break the cell apart, the mRNA comes out. Once the mRNA comes out, you reverse transcribe it. And once you have this reverse transcribed cDNA, you can put this onto a sequencer. How exactly these steps are done across different technology varies, but all the technology that I talk about can be broadly put into these steps of these categories of 5 steps. There are different ways of isolating the single cells. So the field started by literally mouth or micropipetting. There are still 10% of the studies that get published, which actually of how the library gets prepared. So the library preparation can be broadly divided into two categories. One being well-based, which is what I'm highlighting currently here, where you place these cells into individual wells on a 96 well plate. You perform cell lysis, perform reverse transcription, fragment the cDNA and sequence it. There are advantages of doing this is because since you're doing this in a particular well, the cells are physically separate. So there is no way one cell's measurement will mix with the other cell, which will become slightly more apparent as I talk about the second way to do it. So all you do is create the cDNA, fragment it because you cannot really sequence the entire cDNA and put this onto a sequencer. The advantage of using a plate-based method is you get full-length cDNA transcripts, and I'll talk about how exactly in just a bit. The other way to do the library preparation is in the form of droplets, which is where you have the cell along with a gel bead encapsulated in a droplet, and I'll explain what this droplet is and what that gel bead is. What this gel bead essentially does is it carries specific markers for that particular specific barcode for marking that particular cell. You need this because in this case, you don't really have a you don't have a physical separation. All your physical separation is coming from the cell and the bead being in one droplet. Whereas in the plate-based, you have a physical separation. So once you have these droplets, you can barcode the cell. Barcoding a cell means you have a particular primer sequence which sits on the gel bead, which can hybridize with every molecule that is coming out from the cell. Every mRNA molecule that is coming out from the cell can get one unique barcode, which tells you that all the molecules that you're going to measure in this particular droplet came from that one particular cell. Once you do this step, you can then do the rest steps of creating the cDNA and the fragmentation. One challenge with these droplet-based technology, however, is they're not full length. What that full length means is you don't get the entire cDNA sequence. These are generally 3' biased. That is, this hybridization here focuses mostly or tags mostly the poly A tails, so that you get a lot of abundance of transcripts coming from the 3' end of the mRNA, and that makes the 5' end suffer. And I'll show you what I mean by that in just a bit. Okay, so broadly, with these two ways of doing technology figured out, we can just talk about the details of what happens in the plate-based technologies. So in the plate-based technology, the key idea here is to use a template switching oligo, which allows you to sequence the entire cDNA. Once you load the cells into an individual well plate, you have these barcoded primers, which will hybridize with your poly A tail. So you have an oligo DT primer along with a PCR handle, the oligo DT primer will hybridize with the poly A tail. You will add a switch TSO, which stands for template switch oligo, along with an MMLV reverse transcriptase. One of the key properties of this reverse transcriptase is that as it's traveling towards the 5' end of the cDNA, it will end up adding these extra bases, the 3 Cs here. The 3 Cs here will then hybridize with the 3 Gs of the template switch oligo. So the reverse transcriptase, which is moving along this direction, will now switch and will reverse transcribe the remaining strand all the way starting from the other direction. That allows us to profile the entire cDNA, which can then, so obviously you get a cDNA, the cDNAs could run into kilobytes, kilobases of sequences. We cannot really sequence that at once, so we do a fragmentation. The way this fragmentation is done is by adding TN5 transposase. So TN5 is a transposase which you can add Illumina adapters to. What these Illumina adapters are necessary for is for making it amenable to sequencing. So you take the full entire cDNA, you add a TN5 transposase. This will deposit adapters by chunking it off randomly, and this then becomes amenable to sequencing. So what you get out of this is a single cell profile of the entire cDNA in 96 wells at one point. So you can sequence 100 cells, close to 100 cells in one particular experiment. So as you can figure out, this is a laborious process to do because it requires physical separation into wells. But the advantage here is you get the full length cDNA profile. There is a lot of amplification bias that comes because at this step, you require a lot of PCR, again, just because the content of the cDNA is too little for putting it on the sequencer. And as such, the final quantification is prone to this PCR bias. So there are technologies now which get rid of this amplification bias, and that mostly comes from development of droplet-based technologies. The droplet-based technology, the key idea here is to physically separate the cells using droplets. The first paper came out back in 2015, which was a game changer for this field. It was a game changer in the sense because it made this technology really, really scalable. So the key idea behind this technology and the next technology that I'll talk about is very simple. All you want to do is you want to tag individual cells. You don't have a mechanism to physically separate them. You don't want to go to the laborious process of physically doing separation in 96 wells. So you let the microfluidic system do it. What happens in this microfluidic systems is you have two channels, which are one of the channels is giving you the cells. The second channel is giving you the oil. And the third channel is throwing in beads. Now these beads, which I'll talk about and just in the next slide, have primer sequences. This primer sequences will hybridize with the poly A tails of this cell because they get encapsulated in one single droplet. This droplet is formed because there is a T junction here. So the oil and the water ends up making an emulsion. This emulsion causes the cell and the gel bead to encapsulate together. And that gel bead carries one particular barcode, which barcodes the unique nature of that particular cell. Inside this droplet, you will have cell lysis. So the cell will break apart. The mRNA will come out and get hybridized onto the gel beads. Once you get this, once you sort of get that hybrid sequence, you can put this onto a sequencer. So just to give you an idea about what that particular gel bead contains, it has a primer sequence which contains your PCR handle, the cell barcode, and something that's called UMI. So cell barcode will tag unique cell individually. UMI tags, this is a unique molecule which will be different. There are thousands of, if not millions of different UMIs sitting on one single bead. They're meant as a unique identifier for labeling the molecule of cell. The reason you need these UMIs is to avoid PCR duplicates in the quantification step. So it will become clear just in a couple of slides. But what happens once you have this encapsulated? So this is the bead. This gets encapsulated with the cell. You rupture the cell. The mRNA comes out. The poly A tail will hybridize with the poly T sequence here. And then eventually you will pool all the cells at once and carry out the cDNA, the reverse transcript conversion to form the cDNA. So this is broadly, okay, sure, okay. So I have to wrap up in 5 minutes, but I'll just give you a brief idea about what this looks like in the experiment here. So right now what you're seeing is individual droplets coming from this flow chamber. Once this T junction comes here, you form droplets which contain the gel bead and the cell. So I've already explained the steps, so I can in principle skip this. The bead here is a hard bead, so it does not get dissolved. The hybridization happens on the surface. The problem with that, the hybridization happening on the surface, is that you have a lesser number of molecules that are available for hybridization. So I'll just quickly explain what the UMIs is doing. So once you have these genes which are sequenced, you have these reads that come out from this particular experiment. You have a particular cell barcode which tags each individual cell. If you were to focus on these two genes, ACTB, it has the same barcode, so it means it came from the same cell. If you look at the UMI sequence, these two UMI sequences are exactly the same, which is impossible because we want those UMIs to be unique. That's how the bead is designed. What this essentially means is these are PCR duplicates. So even though these are two reads coming from this particular gene, given that their UMIs are similar, we'll count them as only one single molecule. Whereas in this case, you still get two barcodes and two different UMIs. So you get two reads assigned to ARL1, but they're also representative of two molecules. So just to quickly jump on to a different technology, which uses a very similar idea. The only change that now happens in drops, which was also actually introduced in 2015, is that the RT, which is the reverse transcription step, happens within the droplet. So the idea behind using this kind of technology is if you do the reverse transcription within the cell, it enhances the efficiency of RT, thereby you end up losing less of your reagents or wasting less of your reagents. So it's you can talk about different metrics. It's very similar to DropSeq, except that it's slightly more efficient. And there's a commercial version of which merges these two technologies, DropSeq and NDrops. It's called 10x, or the Chromium controller, which you will come across a lot now because a lot of papers right now rely on this particular technology. This technology is very similar to both NDrops and DropSeq, is that it uses a hydrogel which gets dissolved once it gets encapsulated into the mRNA, into the droplet, which causes it to be, which increases the overall reaction efficiency. So why are these technologies 3' biased? So they are all 3' biased because you get a lot of coverage towards the end of the gene body, but less of the content coming from the 5' end of the gene. And the reason this is happening is because there is a poly A selection step. So when you get the read and the barcodes from the gel, the PCR handle explicitly looks for this, but the PCR handle that you have is amplified towards the 3' end because that's what you're targeting. And that's why you end up getting a lot of sequences from the 3' end rather than the 5' end from all these droplet-based technologies. Okay, so just to summarize these different technologies, they all use droplets. There is, if you were to sort of choose and if you had a lot of money, you'd probably go for 10x. Which is the commercial version, which mixes both ideas from DropSeq and NDrops. They essentially rely on same principles, but have different throughput. Okay, very quickly, I'll talk about how to make this happen in individual plates. So the idea, so I'll not play the video, but I'll share the slides and you can play the video on your own. But the general idea here, if you want to do this in plates in high throughput fashion, is to carry out a split pool-based technology. So the way this split pool technology works is it uses plate, but rather than using the physical separation of the cells, of the wells on the plate, it adds barcodes in split pool way. So you add one round of barcode which will get absorbed by the cell. You don't have to physically separate the cells in this case. You add same volume of the solution which contains your cell into the 96 well plate. You add first round of barcode, you remove them, you add second round of barcode into after randomizing the cells or just mixing a solution, and then you do this again. After 3 rounds of barcodes, you have enough barcodes within individual cells so that you can separate this out later on. So it's a cool video which I'll encourage you to watch. I'll share the slides. But just to summarize, single cell technology is broadly classified into two classes, one being plate-based and one being droplet-based. The droplet-based technologies make use of the idea of using droplets as a reaction chamber. And that allows us to sort of get rid of the problem of physically separating the cells. The split pool or the combinatorial indexing technology that I just talked about in the previous slide uses the idea of using the plate, but using the cells as the chamber itself, which makes the advantage of both being almost close to full length, but also being highly scalable and cheaper. Okay, so very quickly, just give, since Professor Sanjeeva talked about proteomics, very quickly in just 2 slides, I'll introduce the idea of doing proteomics in a sequencing fashion. So the idea here is, if you want to profile just the RNA, that's fine. This is what we did using single cell RNA-seq. But can you add another modality onto this? That by another modality, can you actually also measure the protein levels or not? So this was technology that was developed where I work at. It's called CiteSeq, which allows you to profile antibody, which allows you to profile surface proteins by using the simple idea that you can conjugate the antibody that you want to, which is the antibody for the protein of interest, along with an oligonucleotide. This oligonucleotide has a PCR handle along with this poly A tail. So once you have this poly A tail and the barcode and the antibody, you can use any droplet-based technology. What this droplet-based technology will then do is, since you have this poly A tail, it will get hybridized to the gel bead, similar to what was happening in case if you were to just measure the RNA. What this allows you to do is it allows you to basically measure both surface level proteins and single cell RNA-seq all at once in one experiment. You can do this in 10x, you can do this in DropSeq, or any based of droplet technology. So that allows us to brings us to the wrap of this presentation. There's just one more modality that you can add, which is the open chromatin regions, which is where you can profile a single cell attack-seq, which is a way to measure which regions in your DNA are open. The way this works is a very simple reaction. You have the open chromatin, which will get targeted by the TN5. The TN5 will deposit its own primer sequences, which can then be selected, and you can do this in single cell. And you can, what this single cell attack-seq allows you to do is that it allows you to ask questions about how is the chromatin change reflected in the expression change. So you can ask questions about which motifs are open and how does that relate to changes in the gene expression, and whether this particular differentially accessible region is the reason a particular gene is highly or lowly expressed. And finally, this is my final slide. We have this technology called dogma-seq, which literally starts from where I started, which is the central dogma, which allows you to profile in a high throughput fashion in droplets, open chromatin regions, RNA and protein all at once. Yeah, thank you.
Thank you, sir. It was really interesting to know all these new technologies.
Is there any question?
Thank you. That's a really interesting presentation, very clear. I'm quite interested in the models phenotype in cells.
Mm-hmm.
So if you have a pool of single cell RNA-seq, and for example, you wanted to identify cells which were undergoing senescence in your pool, how many models exist out there for those? And how reliable are they?
Great question. So I don't have a slide, but I'll just explain what we usually do for figuring out what is the cellular state. The idea here is to utilize the prior information that we really know about genes. So there are certain genes which will go up in terms of if you are in G1 phase, G2 phase, or senescence. If we use those genes, we track their expression pattern, and we can quantify what is the relative expression pattern of those genes which are associated with, let's say, cell cycle phase 1, cell cycle or G1 phase or G2M phase or G0 phase. We score those particular genes, and then we come up with a classification that this cell particularly belongs to G1 or G2 or G0. So the idea is, if you have a prior knowledge about which genes are involved in one of the pathways, you can score them at the cellular at the single cell level, and then quantify which cell goes to which of these categories.
Could you turn that back on itself as well? Because that's giving it a code, the definition of the cell based upon a prior hypothesis.
Mm-hmm.
Could you extend that hypothesis by then turning it back on itself?
Wonderful question. And yes, the short answer is yes. And the way you can do this is because you have information about other genes in that cell as well. So what you do, for example, in one of the projects, what I'm currently doing is doing exactly what you're talking about, is we don't a priori know what signature should mark cells that are going into senescence. In this case, this happens to be glial cells that I work with. So the way I do this is I start with the prior knowledge, figure out a score, and then once I get the score for those cells, I look for what is so heterogeneous or what is similar between those cells versus the rest of the cells. And that can help you come up with a unique signature that is not dependent on prior knowledge, that helps you differentiate one population from the other. Any other questions? I'll be so
we can take in a follow-up.
Sure. Okay.
We'll
Sure.
have a hands-on.
Yeah, I have a hands-on, so yeah, happy to take any questions that time. Thank you.
So to express our gratitude, we'd like to felicitate Dr. Saket Chaudhary.
So let me just add here. So Dr. Saket Chaudhary now, going to be assistant professor in one of the IITs very soon. Same area with programming skill and continue to work in all this field. And also specialize on this particular cutting edge field, which is upcoming for the RNA-seq and protein synthesis, what we are talking right now. So we are very proud to have students like him. And if there are some undergrads in the crowd, I think you should think about how best you can leverage some of these interactions and then take those knowledge forward and be the leader in your field. So congratulations, Saket, on all your accomplishments.
It is my pleasure to welcome Dr. Jaya Goyal on stage. She's a researcher, practitioner, specialist with 16 years of leadership experience in research management and administration, policy evaluation in higher education in government, academia, health and nutrition sectors. A thought leader creating products, programs, critiques and content on science diplomacy, social policy and higher education policy. Jaya is an ex-faculty and alumnus of Tata Institute of Social Sciences, Mumbai, ex-researcher at London School of Economics and Political Sciences, ex-head higher education at British Council India, ex-senior consultant at USA, Government of Maharashtra, and ex-junior fellow at Observer Research Foundation, Mumbai. Jaya has a PhD in social sciences and MSc in Human Ecology and Community Nutrition. She is the co-editor of a Macmillan published book titled Mumbai Vision 2015, an agenda for urban renewal. In 2007, running in its third edition. Now, she has also written a monograph which is titled Public Services Delivery, Plus Accountability and Local Institutions Mind. It is sponsored by Indian Council for Social Science Research and is due for release soon. So, looking forward to your talk, ma'am.
While the screen is being set up, I would definitely not call myself a misfit here in the crowd full of bioscientists, engineers, probably data scientists, computer scientists, probably because I believe that the silos in sciences are created for largely operational purposes, but eventually, if you look at any problem, everything converges. So I thank firstly Professor Sanjeeva, Professor Graham, for extending this invitation to me to speak at the Monsoon Advanced Proteomics School. And I hope by end of this talk, you would feel a little bit more connected with me, and that there would not be too much of disconnect, because I'm a social scientist here, and I'll try and connect with you with the work that I have recently done. So with that, I will be speaking today, as the slide deck is coming. I'll be speaking on the work of building stereotypes with large language models using community engagement for scale and depth. So as a social scientist, how I could actually collaborate with computer scientists, with linguists, with data scientists who work on big data, to come up with the project, and why did we come up with the project. So the title of my presentation today is Building Stereotypes with Large Language Models and Community Engagement for Scale and Depth. And this presentation was actually created by the team of researchers, which included Google research from California, Google India. It had researchers from Virginia Tech in the US and Sigardian Connect. I am the founder director of Sigardian Connect, a research firm, and it was my first year of my starting as a research startup. So this presentation was first given by one of our team members at an international conference on the European chapter of Association of Computational Linguistics on 4th of May 2023. So it's a very recent work, just a year old. We've already got 2 papers out of it. One of the papers is this. The presentation, of course, was created by the Google team, so I give them the full credit of creating this presentation. And the paper, of course, is under a publication in an international journal. So in this work, we discuss pathways towards building repository for evaluation of the harms of stereotyping. We are all aware of how internet is every day, too much controversy is happening, and there is a large amount of content getting generated with a lot of language-based violence that can happen through use of media, Twitter, and now we have so many social media channels. So this work was, of course, this work relates to the large language models, but why was it the need for having a community engagement, and why would a social scientist get involved with large language models? So that's what this presentation is going to answer today. So we focus on ways to do this at scale, because it's just not about one region or one state or one country. It's about the large World Wide Web. So how do you do this on scale, along with capturing different regional nuances, because we have thousands of languages, cultures, geographies. So how do you do it across regional nuances, complementing different strategies, and partner human experiences with capabilities of the large language models?
Yeah.
Okay, so the next slide. Yeah, thank you. So before we go on to the work, it's important to note that some of the content in the slides can be offensive and it can be triggering. So I'm already warning you, but of course this is something that that was the whole idea about what is it that is coming up as stereotypes. Next slide. Yeah. The current problem or the question that we ask in this work is, how do we robustly build and scale evaluations of the harm of stereotyping, such that they are representative of different cultures, global cultures, and societies. The problem can be seen as layered because first, there are already limited resources. So obviously no other, it's about Google, of course, it's about Google, but even they would not have, unlimited resources to do such body of work in every region, every state, every language. So how do you do it on scale? So that was one of the key questions that we were trying to grapple with. And if you see, the problem is layered because firstly, there are limited resources, as I said, on stereotypes present globally. And a lot of work focuses on stereotypes seen in the West, especially in North America. So there is a good body of work, but it largely focuses on the global North. Next slide. As we've seen in this plot, this covers data from 6 different predominantly used resources. The number of stereotypes known globally and the coverage of the globe overall is low in sub-Saharan Africa, Latin America, and particularly with very, very little coverage. So there's hardly any resources available to study stereotypes in these regions. Yes. So as we, it's very clear that the major issue with these evaluations is how we have used Western lenses all the time, Western narratives of fairness, of disparities, and stereotypes about the rest of the world are often more Western produced rather than stereotypes commonly present in these specific regions. Issues such as fairness and you would know that issues such as fairness and ethics are inherently socially constructed. So this is especially problematic for interventions framed solely for the Western context. So largely Western context may use, I'll give you examples later on, but for all Asians or for all Indians, they will use the word brown. So there is no granularity about, Indians are so ethnically and linguistically so diverse, but they will paint every, either they'll call them Asian or they'll call them brown. So that is a Western concept, Western stereotype, so there is no, I would say, no further bifurcations of what brown, and brown itself is a stereotype because it gives us skin color. So these were some of the issues that we were facing with. The next slide, please. Okay. So it was very vital to bring local communities and their perspectives about the axes of disparities that are unique, culturally situated within that region in order to build a very nuanced evaluation paradigms that are able to solve these diverse global contexts. Next, please. And for this reason, we devised complementary approaches to leverage generative AI and engage with local communities which together can bring both granular coverage of global identities as well as immense scale and depth. So we wanted the scale and we wanted the depth, both at the same time. Okay, so if we imagine, as you can see on the slide, if we imagine that there is a universal set of stereotypes, and this Venn diagram shows, and as a lens, if we thought of as a society or a window to the society, then these existing models are trained in vast amount of textured data. They do a lot of learning through social information, and then they are able to generate some substantial fractions of these stereotypes. But not all generations are stereotypical and there can be a lot of noise in the data. So what if we only take the data generated on the internet and just use those to build repositories of stereotypes? The issue with those data is that there is a lot of noise and then it cannot be representative because we cannot assume a large population to be on the internet. So those were some of the issues because of which we have to situate them through human validations of this entire data. Next slide, please. Yes. So to this end, we developed a stereotype benchmark with broad coverage by partnering human knowledge with generative abilities of NLMs. NLMs would be natural language processing models, and we started by using human validated stereotypes from existing resources. So we start with existing resources. We went through what are the existing stereotypes and used them as a prompts to collect the data which we did in this work. So there was the existing resources which included UNESCO data and some NLP stereotype benchmarks such as crossbars and stereoset. We use the seeds in a short prompting setup. So we gave prompts like Sindhis are traders, for instance, because this was very clearly documented. Or Brahmins are vegetarians, or Jains are vegetarians, things like that. And those prompts which were already documented, these prompts were already existing in the current data. We use these existing prompts to collect more data on stereotypes. So the smallest data generated contains stereotypes as well as noise, making our subsequent step of human validations extremely important. So each and every stereotype collected were also validated and anodized. We structure this particular step extremely carefully to reflect socio-cultural context. Yes, socio-cultural context, and essentially we saw that we obtained annotations for each stereotype from annoters that were identifying or living with this. So a person who is looking at stereotypes to say whether they're valid or not cannot be a person from a Western background. They had to be people from that particular geography. So this pipeline gave us a lot of, I would say, advantages. Most of it is that we now have much greater coverage of stereotypes globally, and with over 10 times the identities covered as compared to human-only generated benchmarks. So as we see on the plot here, it's just not scaling up, but also scaling out. So I believe you all are in science research that is more reductionist. So you go down at the cellular level. But in this work, we actually zoomed out and we saw what is a macro picture. How is this the big data in linguistic looking like? So it is just not scaling up, so scaling out and broadening the coverage across the globe with large coverage of the global south in particular, including Latin America, and that's what the study was based out of India. So it was the first study that Google kicked off to understand and collect data on social stereotypes in India. I'll just briefly come back to it. What did we do here? But essentially, in this Venn diagram, you can see that we talked about how LLMs, large language models, can be a lens in the society, and using human validation to weed out spurious associations, we came across this resource. However, LLMs do not give us a complete picture of the society. So why did Google actually thought that whatever data they have been generating out of LLMs, that is not giving them the real picture, and why do they need to actually go out in the community and collect more data? Because it was a skewed perspective, shaped by what kinds of things are discussed online, as I said earlier, and what sources these models were using for training. So on the right side, if you can just stay on this slide, on the right side, you said the social context was obviously missing in these large language models, and that's how the study filled that gap. Next slide, please. So yeah, we recognized this and adopted a community-based collection approach towards stereotype resource building in India. And with this engagement, our team of researchers, which included, as I said earlier, about Google research team, along with me as a social scientist with local expertise, and having worked with marginalized communities in India for over a decade, we ensured diversity of participants. So we took students essentially studying in public funded policy-based affirmative action or reservation policy, so that we can take in respondents who are from diverse backgrounds. So that is what we did, and we ensured diversity and by gaining responses from these students, and that's how we were able to collect the data. The data collected through this engagement has covered a lot of crucial insights that we would have missed out if we relied only on large language models. First, this work, next slide please. Yes, first this work captured a very fine-grained intersectional identity groups, for example, Dalit women. And this combines both, as caste and gender, and it is considered as, of course, it's considered as one of the most, I would say, marginalized communities in India. So things like that. So we got more nuanced understanding of what it would be like to either create stereotypes of have stereotypes of Dalit who's a woman as well. So things like that. Second, it has captured a lot of complementary resources or stereotypes to large language models generated pipeline, and it has a very low overlap with it. So much of the data collected was very unique and it didn't have too much overlaps, which means there was so much gap in existing data, which this data could fill it. And third, it complements the Western lens, which is prevalent in many other resources. So for example, attributes of vegetarianism are brownness or dark skin, as I said earlier, were associated in a blanket way to all Indians, and which was broken down into many other granularities. Next slide, please. The data also brought in a lot of colloquial terms like Banyas or Sindhis and associations. So a lot of, I would say, occupational groups, making it very socially relevant in the Indian context. On the other hand, the community-based, effort has been more focused in India. Does not really, have stereotypes for the rest of the world, much as the LLM method does. So, if you see here, association of being good in math gets broken down into many different types by the generative method. So it's here that the community engagement models give us very rich stereotype associations. My final slide. Overall, this entire research led to building complementary resources. And with this work, we demonstrated how to do this on scale and how to contextualize these benchmarks in terms of coverage in global context and around the world. We demonstrate specifically how these pathways based on generations and community engagement can be imagined to be complementary to each other and used alternatively. One is providing a granular seed stereotypes to scale up with, and the other is providing insights with what are the major gaps that needs to be filled with community engagement. So these subtle and implicit characteristics that were discovered helped us to create and establish a very effective evaluation standards which are socially, culturally inclusive and sensitive. So I'm through with this with the slide, but this of course will not give you a full view of the data that was collected, because most people are interested, okay, what were the stereotypes and what was the data, because this release of these stereotypes will happen publicly by Google. And we also have a second paper that actually talks about these examples of social stereotypes in India, and that paper is going to get published very soon. So before that, I think it's not ethical for me to share it at a public platform like this, but this would be a publicly available data for researchers to work on. So thank you so much. I hope you liked and enjoyed this presentation, and I'll be very happy to answer any questions. Thank you.
All right, so it is wonderful to hear how the big data is for the social context, and that was our vision to that we are all generating data, but how it has to be meaningfully reach to society. And the remaining way, I think we have to have the social scientists now together with the big data scientists. And I think Raja was mentioning that BSc is now considering to having all the projects on social scientists as a must entity to be involved, which I think will be very relevant. So we will take a lunch break, and during lunch, I think let's take a question that time because we are running a bit short of the time. So we will have a discussion with Dr. Jaya over the lunch. Please, talk to her. She brings a lot of extensive experience working in different industry domain, including British Council. So do utilize, her time and all the experience here. Thank you very much.
Thank you. Thank you so much.
Thank you, ma'am. We would like to participate in the booking.
It was wonderful. Thank you so much. My pleasure. My pleasure.
Proteomics is quite decent in terms of the Gaussian pattern, right? You'd like to which one?
Yeah, this one. Yeah.
So you'd like to see that the peptides have come out of the column and now it has become flat. It means there is not much carryover. Other thing you look for the intensity is very high, which is 8 power 9 or 10. If it goes between 7 or 8, then it's not so good intensity. And if now I think Avinash is highlighting the MS fragmentation. And for each, MS fragmentation, for each MS, what is the MS number of spectra being, MS?
More than 30.
So for each element, almost 30 or so MS fragmentation happens. So now this is good enough for you to get the sequence coverage, right? This is all you do in the mass spec, right? Just everything comes to you on the data, and you're seeing the frag file, how to analyze it, and then we can now take it forward. So this is typical kind of, the LFQ and level 3 quantification. All right, let's continue talking over the lunch, okay? And be back by 2 p.m. Thank you.
unmute. Sorry, I'll share like this. Is it visible on Teams? Is the screen visible on Teams? Is it visible on Teams? Not yet. Yeah, so if I do this, will it? Okay, it's fine. Yeah, that also works. Okay. Okay, so for everyone who's also online, since you already have internet, and if you have R and R Studio downloaded, you should be good to go, except that you also require an object. So if you go to this link, there's a header which says download the object, which has the link to the object. If for some reason your internet is not working, so if you navigate all the way to bottom, if you click on this link, you should be able to download the object. If for some reason you don't have internet, in the room there is a pen drive going on which has an RDS file, which is the object which you can get from this link as well. So make sure you copy that somewhere in your directory. Just to make sure, does everyone have your R Studio involved installed and any issues there?
If they are facing any errors in the answer.
Okay, so there are people here, volunteers here who will be more than happy to help you with installation issues with R Studio or the packages. It's also okay if you cannot install all the packages, though hopefully you should not have any trouble. They're straightforward to install. So I'll not walk over the instructions here because they're very limited, assuming you have already done R and R Studio installation already. If you have not installed if you have not installed R Studio, then you need to do this before we move on to the next step. But at this point, is it safe to assume everyone in the room has R Studio open and working? If not, please raise your hand and very happy to help you. Okay. Okay, so this is my R Studio window. What I have open here is a notebook, analysis.rnd, which has both text and all the software, all the code that you will require for running this vignette. It's called a vignette. It has all the instructions that you require. You can also see this online by clicking on this. At the bottom towards, there's an analysis notebook link, which if you open will, sorry, I cannot change. Okay, yeah. Which if you open, you can navigate this online as well. It's an HTML file. So at the end of the workshop, the goal here would be that you reproduce this at your end totally. And I'll walk over each and every step that we are doing, why we are doing it. It's also, the rationale is also explained in this vignette. So for whatever reason, if you're not able to complete, like don't be disheartened, you have all the information in this notebook. So you can always go back, be able to run this from anywhere. Once you have the object, no internet required, so don't worry about that as well. You should be able to run it even on your flight or train back home. Okay. Okay, so I'm assuming everybody has R Studio involved. If not, now is the right time to pause me, because once we start, we'll keep going. Yes. Astrid, can you help me with Astrid? So I'll wait for a couple of more minutes.
We have Orco and Sabyasachi over there who will be helping you if you are facing any issue in the art studio right now.
If you want the link so the link is very simple, bit.ly /maps2023sc for the instructions. If you open that page, everything that you need for this workshop is on that one page, including all the links, what you need to do to get started. Okay. And I start. Okay. So what we are working today is this paper, so I'll switch to, I'll keep switching between presentation, but this paper came out a couple of years ago. It's a fairly straightforward paper in what the analysis tries to do. The paper is asking a very simple question. What changes between an Alzheimer's brain sample as respect to a normal wild type sample? This data set has both human and mouse data sets. Human data set for this particular sample, I particularly did not want to use because it's tricky in this case to circulate that particular object for ethical concerns. So we work with the mouse analog, and the simple way to do this is the object that I shared with you. So there were two